We are live on YouTube.
You can start.
Are we good to go?
Yeah.
Yes, Shilpa madam, we can go. We can go. Good to go. Yes.
Vakratunda Mahakaya, Koti Surya Samaprabha, Nirvignam Kurume, Karyeshu Sarvada. A very good morning to you all, and thank you for joining us all today. On behalf of the organizing committee, Sakura Science Forum, Nitte deemed to be University, and University of Miyazaki, Japan, I, Dr. Shilpa, your host for the day, extend to you all a very warm welcome to the International One-Day Virtual Conference on COVID-19 Pandemic, yesterday, today, and tomorrow. The coronavirus pandemic has caused enormous damage to health, wealth, and well-being, like a world war, except in this case, we are all on the same side. Everyone can work together to learn about the disease and develop tools to fight it, as global innovation is the only key for limiting the damage. With innovations in testing, treatments, vaccines, and policies to limit the spread while minimizing the damage to economies and well-being, the national conversation has now turned to a single question: when can we get back to the normal life? These were the words from the Bill Gates blog, Gates Notes, and this is analogous to today's discussion. Now, without any further ado, we will turn the time over to the welcome address by Professor Dr. Fatima De Silva, Chairperson, Nitte Sakura Science Forum. Over to you, ma'am.
Warm greetings to all from Nitte deemed to be University. As the entire nation is grappling to fight COVID-19, collaborative efforts between nations, organizations, and communities have proved to be our biggest strength to get through this pandemic successfully. The importance of collaboration through exchange of best practices and solutions across the globe is thus well asserted. One such fruitful collaboration, founded in 2019, has been our very own Nitte Sakura Science Forum. This novel initiative of our Honorable Vice Chancellor has proved a unique platform for us today to share valuable ideas through speakers from Japan, UK, USA, and India. The discussion today would open up a new dimension on dealing with the challenges that COVID-19 has posed, and I, Dr. Fatima De Silva, take honor to present to all the welcome note. A hearty welcome to our Honorable Vice Chancellor, Dr. Satish Kumar Bhandari, our pillar of great strength and inspiration. A warm welcome to you, sir. It's my great privilege to welcome Dr. Harish Madhyastha, Associate Professor, Faculty of Medicine from University of Miyazaki, Japan, who has been the driving force in founding the Sakura Science Forum. A hearty welcome to you, sir. We are fortunate to have two eminent scholars, Dr. S.P. Srinivas, Professor at the Department of Pharmacology, Indiana University, USA, and Dr. Vimal Karani, Associate Professor in Nutrigenetics and Nutrigenomics from the University of Reading, UK. Welcome, dear professors. Today we would also be hearing personal experiences of our frontline warriors in the task force, our esteemed doctors from Shema, Dr. Ganesh, Dr. Amit, and Dr. Giridhar in a panel discussion which would be moderated by Dr. Vadish Bhatt. And finally, I take this opportunity to extend a cordial welcome to all the enthusiastic participants in this webinar today. Wish you all a pleasant morning and a very insightful conference ahead. Thank you.
Thank you, ma'am, for that very warm welcome. May I now request our Honorable Vice Chancellor, Professor Dr. Satish Kumar Bhandari, the man with a distinct vision and inspiration for all of us, a strong supporter for all the research-related activities, to give away the presidential address.
Very good morning. My good friend, Professor Harish Madhyastha, the resource persons, Dr. S.P. Srinivas and Dr. Vimal Karani, President of the Sakura Science Forum of the University, Dr. Fatima, the main architect of this whole program, Professor Sucheta Kumari, Other resources from my colleagues from the university, office bearers of Sakura Science Forum, and my dear participants of this prestigious day conference. As you all are aware, we are going through an unprecedented global health crisis due to COVID-19. As explained by the previous speakers, every section of the people, it may be from education, industry, tourism, have been affected, and there's a huge amount of job loss, and people are facing unimaginable hardship throughout the world. COVID-19, the term coined by WHO on February 11th, 2020, for this Wuhan virus, took the world by surprise and causing, we do not know how long, misery and instability and pain to the people and around the world. Everybody is suffering due to this virus. It has affected rich and poor, white and black, princesses, prime ministers, and ministers have been affected by this virus. So the bubonic plague, smallpox, Spanish flu, and other infectious diseases have devastated mankind before. But like COVID-19, mankind has not experienced before in terms of magnitude of the problems. When the lockdown was announced by our Honorable Prime Minister on the 22nd of March, the first Janata lockdown and then regular lockdown from 25th, it came as a bolt out of the blue for most of the people, including the university. But we should congratulate the people of our country for cooperating with the government, for implementing the lockdown guidelines very religiously. Yes, there were problems. There was a lot of job loss, hunger, migration of the laborers, reverse migration, shortage of masks and PPEs. But we managed it to a large extent by the cooperation of the people, particularly the poor, marginalized people, doctors, nurses, paramedics, scientists, and the government solidly behind the people. Compared to most developed nations, we have done fairly well in terms of mortality and recovery rate. Today, we are self-sufficient in masks, PPEs, ventilators, and hospital bed. There is adequate food to be distributed through public distribution system, free of cost. But we cannot be complacent. Unfortunately, fatigue has set in amongst the people. Relaxation of COVID guidelines, coupled with the careless attitude of the people, likely to cause a second wave. This can be devastating in terms of morbidity and mortality in a nation with 1.36 billion people, for which may have to pay a very huge price. With specific treatment and vaccine for this ill-understood virus, still a distant possibility, the only way is to cope with the virus and live with it, following strict guidelines, and also to educate the people about the measures to be taken to contain the spread of this very funny virus. I am proud to tell you that our university has taken certain proactive measures from the very beginning. Successfully implemented the blended learning and evaluation process in all its constituent colleges, and the first and final examination of all courses of the university have been completed, fully abiding by the government guidelines. Thousands and odd undergraduate and postgraduate and PhD students will be awarded degree in the forthcoming convocation, which has been fixed for 9th October this year. The new academic year for intermediary batches have been already started in the online mode, and new postgraduate batches already joined again in the online mode. Our 1,000-bed tertiary care hospital has dedicated 500 beds to COVID-19 patients with very strict standard operating procedures. University has started COVID-19 diagnostic lab and plasma therapy unit in the hospital, and it's helping the government in the government hospital to contain this pandemic effectively. Now coming to today's program, I'm glad that our Sakura Science Forum, which was started by the university for collaborative research and education with Miyazaki University of Japan, has organized this unique conference in virtual format on COVID-19 yesterday, today, and tomorrow. I congratulate the office bearers of Sakura Science Forum for organizing this one-day conference. And according to me, the theme is very appropriate and very timely. I thank Professor Madhyastha, our son of the soil, researcher and professor par excellence from Miyazaki University, for his help in establishing memorandum of understanding with the Miyazaki University, and also my organizing team for organizing this conference. Madhyastha was responsible for the student exchange through JST Sakura program between the two universities. I also recognize the contribution of Professor Ikanoue, the president of Miyazaki University, and all senior functionaries of the Miyazaki University. Also, Professor Radha Madhyastha for the successful implementation of various objectives of this understanding between these two prestigious universities. I have gone through the program highlights of this conference. I have no doubt that this will be a very successful conference as far as the content and delivery of this topic is concerned, under the leadership of Professor Sujeta Kumari, Professor Fatima, Professor Harish, and other very eminent resource persons and office bearers of Sakura Science Forum. I hope the participants will have very fruitful academic deliberation, and I wish grand success to the international conference in a virtual format. All the best. Thank you for the opportunity given to me. Thank you.
Thank you, sir, for that thought-provoking and informative address. Today, we have with us the flag bearer of the Nitte Sakura Science Exchange Program, Dr. Harish Kumar Madhyastha, who will be delivering the inaugural address. But before that, it is my pleasing duty to introduce Dr. Harish Kumar Madhyastha. Dr. Harish Kumar Madhyastha's academic journey started with a BSc degree from Mangaluru University, Masters from Saurashtra University, Gujarat, and PhD degree in Applied Sciences from Sardar Patel University, Gujarat. Later on, he also obtained his second PhD degree in molecular medicine from the University of Miyazaki, Japan. He started his career as a scientist in Sri Murugappa Chettiar Research Center, Chennai, India, where he worked on single-cell protein cultivation technology under the able leadership of Professor C.V. Sheshadri, which was subsequently expanded to major spirulina-producing industries, namely Paris Spirulina, Chennai. Later on, he pursued postdoctoral research at Miyazaki University that culminated in a faculty position in applied physiology at the University of Miyazaki. Generation of novel platforms for delivery of nano-sized biomaterial payloads into specific cell types and the utilization of this smart model as a new drug delivery system for wound healing are some of his research interests. As a nutraceutical biomaterial for wound healing, he formulated nano-sized phycocyanin, curcumin, venophalon, emodin, quercetin as selective cell migration regulating agents and other biomedical applications. As a member of several projects, he is deeply pondering to understand the role of nanocargo exosomes in late keratosis of arsenic toxicity in human. His academic credentials are credited with H value of 22, Clay-Variate Analytic Cumulative Impact Factor of 103.5, and a RG score of 30.1 with 6 international patents. He is also actively involved in many international projects, including ongoing Indo-Japan scientific and academic collaborations. He is also shouldering the additional responsibilities as Indo-Japan academic spokesperson of University of Miyazaki to further strengthen the cohesive relationship between India academicians and University of Miyazaki. I once again welcome to you, sir. Now over to you, sir, for the inaugural address.
Yeah. So, thank you everybody. Shubhadin Pranam. Respected Chancellor, Vice Chancellor, Pro-Chancellor, Pro-VC, DG Research, Deans, Head of Central Research Lab, faculties, my dear students and Pan-India participants who registered for this virtual international conference. Thanks a lot for giving me an opportunity to share my thoughts on this inaugural address. It gives me immense pleasure to present in the Sakura Science Forum a brainchild spin-off of Nitte University, being a STEM, STEMM university, a pleasant place of learning with transdisciplinary thoughts and dynamic management. I think we should dedicate today's event to COVID frontline warriors like doctors, nurses, paramedical staffs, and ground heroes who selflessly serve the society. They deserve a big applause. Topic of today's conference, COVID-19 pandemic, past, present, and future is truly innovative. Yes, year 2020 is marked as a COVID year. It is sad and horrible journey of virus from Wuhan city to entire world. Maybe the first of its kind after the genesis of the earth. Just with the 9-month yatra of this virus, entire world has been shaken completely with negative growth of economy. Every government, as we know, is taking a severe action to tackle, but virus outsmarts the entire human decision. Fine, what's the next? Just yesterday, Professor Anthony Fauci, head CDC America, warned that this pandemic may run for another 1 year. Meanwhile, we scientists took this as a challenging task to come up with many ideathons to heal the world. I had a chance to interact with some of the policymakers of Government of India in Indian Embassy at Tokyo. Their ideas and foresights are really appreciating. In India, in last 70 years, there is an impending suggestion for any translation research with a default slogan of Aam Aadmi. But now, policymakers reformed with thoughts of practical application, which is the idea of startup. I have been giving a bio-inspired ideathonic hypothetical talks in various IITs, ICERs, NITs, national universities, and private universities on this bio-inspired ideathonic talks, including Nitya University last year, where I told in one day all school children should go to school with a mask. This was the talk of 3 years before in IIT Kanpur. But some people took it as a igniting idea. Now result is coming. Swasa 95 mask of IIT Kanpur is the first mask of India which is available in the market now. A welcoming side of development. So if you think what's the future, here are some of the suggestions. One, water jacketed countries are better immunized than the land bordered nations. So do we have data on COVID-19 rate of infection, rate of death or recovery in coastal villages of India? I think Nitya can take up this project as a small master's dissertation topic. Second, herd immunity is promising sign of further research, like neutralizing antibody techniques with Pac-Man component may give some relief sign. Third, social lifestyle is another avenue for the research. For example, if you see in South Africa, there are 631,000 cases from March onwards, but only 14,879 deaths, 7 times lower than the world average. Any clue? Fourth one is symbiotic action of prebiotics and probiotics. So this is all biotics engineering. I think we should think about it. Fifth one is studies suggested that human receptor for COVID-19 may be an angiotensin converting enzyme 2, H2 receptor, similar to that of SARS-CoV. The nucleoscapid protein of the COVID-19 has 90% amino acid sequence identity similarity with the SARS-CoV. So that we should do something on this particular receptors. Sixth one is chemotoxins for the biopoisons. Do we have any severe poison chemicals to tackle this virus? We can think about this. However, there are so many ideas which are available in the world and the scientists, including Nitya University professors and labs and other people. However, today we have two eminent speakers from USA, UK for their enlightening talk would certainly give us a novel platform for further research activity and joint collaborative actions. Let's listen to them. So I once again thank all management, professors. Special thanks for my good friend, Professor Satish Kumar Bhandari, Vice Chancellor, where we have collaboration research since last 3 years. Thank you, one and all. Have a good day. Bye.
Thank you, sir, for that intriguing inaugural address. Moving forward, today we have with us two eminent speakers for the day, Dr. S.P. Srinivas, Associate Professor, Indiana University, USA, who will be deliberating on the topic Pharmacological Approaches Against COVID-19, a review of current knowledge, and Dr. Vimal Karani, Associate Professor in Nutrigenetics, Nutrigenomics, University of Reading, UK, who will be deliberating on topic nutrigenetics and cardiometabolic diseases and focus on COVID-19 and nutrition security. The moderator for the session is Dr. Harish Kumar Madhyastha. So may I now request Dr. Harish Kumar Madhyastha to take over the session and introduce our first speaker for the day, Dr. S.P. Srinivas.
So hello, once again Harish here. I am pleasing to introduce Dr. S.P. Srinivas, Professor of Indiana University School of Pharmacy. Many of them may not be knowing he is a son of Kannada. He is a Kannadiga. His specialties is a transcorneal drug diffusion, physiology of cornea, pharmacology of trabecular mass-wax outflow, barrier integrity of ocular epithelia, the formation of adherents and tight junctions of the cells, nanoparticle for the drug delivery and biosensing, response to the pro-inflammatory cytokines. He has received his PhD in chemical engineering from Drexel University in 1987, consequently postdoctoral training in the Stanford University in the area of ocular drug delivery, then University of California, Berkeley in the area of corneal physiology. He is IIT graduates and with chemical engineering. He taught systematic pharmacology and pharmacology as a teaching professions. He has several publications in the high impact fact journals, and of course, I am one of the co-author of several of his publications also. And the current research includes the regulation of barrier integrity, corneal endothelium, regulation of aqueous humor outflow from trabecular mass-wax cells, and pharmacokinetics of topical drugs to eye. He has more than 50 reputed international publications in the national and international journals. Over to you, sir.
Thank you very much. Hope you can see my screen.
Yes.
Let me project it, yeah, and move the screen out here. Okay, can you see my screen, please?
Yes.
Okay, thank you. I thank you for this opportunity, and especially the organizers. Dr. Madhyastha approached me first, and then later Dr. Sucheta Kumari. And I appreciate the remarks by Dr. Bhandari and all the others. And I'm given to science, so let me get directly to the topic, COVID-19 pharmacology. And as you know, the COVID-19 is the disease. I have roughly 60 slides. I want to sort of briskly walk through certain slides, but the idea is to give a big picture of where we stand today. To be very specific, I was taken, I sort of like this very first sentence I saw in a publication by Garvin et al. In this month's issue of eLife, and they say neither the disease mechanism nor the treatments for COVID-19 are currently known. That is the state of affairs. We don't know the disease. We don't understand COVID-19 in its entirety, and also we don't understand how to treat. There's no magic bullet at the moment to treat the disease. Especially, as you know, the disease manifests in 3 phases. We call what is called asymptomatic phase, and then symptomatic phase, and then there is this so-called hyperinflammatory state, which is the most devastating. In fact, we don't have the major problem comes about with this virus, which is SARS-CoV-2. This is the severe acute respiratory syndrome coronavirus 2, which is the causative agent for COVID-19. We know many coronaviruses, but this coronavirus comes gifted with certain characteristics. We'll try to highlight them. But what is important is that it has many characteristics of SARS-CoV that which came out from Guangdong province in China in 2002. And there are some similarities to MERS-CoV as well. At least they are all coronaviruses. But this disease, this coronavirus 2 is contagious and is causing beyond simply pneumonia, beyond what we call ARDS, ARDS. And it is causing multi-organ damage. And we don't have any, we don't understand the pathophysiology. There is no drug really to treat, let alone the hyperinflammatory states. But we also don't know how to treat even the multiplication of the virus. Okay, that's where we stand. So, of course, we are all hoping that the vaccine would occur very shortly, and that's the major focus we should focus. We should be driving it. Okay, with that said, so I have to thank my students who helped me compile these slides. Two students from computer science, Bhavya and Vishaka, then two medical students, Kavit and Stuti. And so I have 11 points to make. First, let's try to talk briefly about how SARS-CoV-2 is different from coronaviruses. Then let's focus on the spike protein. Remember, the spike protein of these coronaviruses, they are the main antigens of this virus, and they are also the way they use this spike protein to enter the human cells, the target cells in the human body. And let's quickly go through the virus life cycle, because most of the drugs are, we are trying to develop are targeted towards the virus life cycle because we want to curb viral multiplication. And we will briefly touch upon viral transmission and then discuss within a single slide or so clinical features of COVID-19. Then we'll see how the immune response is orchestrated, because after all, what is happening is in many people, especially hospitalized people, we have this overdrive of the immune response leading to multi-organ damage. At least that's one of the reasons. And so let's try to focus on that a bit quickly to appreciate that the cytokines are produced, how it is orchestrated, how the immune response is bigger, and what are the complications that we see, and especially in the context of multi-organ damage. Okay, and what is the pathophysiology of the septic shock? That is the underlying the so-called many of the symptoms of COVID-19, which of course our doctors will discuss very soon. And especially we should focus on what we call cytokine storm, and recently what has been pointed out as bradykinin storm. Let's see how the bradykinin storm could be underlying so-called septic shock that we see in many patients who essentially succumb to death. And after all this background, we can quickly go through, think of pharmacological targets in the form of targeting the virus itself, specifically these aspects of the virus, that is the viral entry and the viral proteases, and then the viral RNA-dependent RNA polymerase. Then we'll talk about how to handle the cytokines, and the cytokine storm and the related consequences on the host, that is on the human body. Then, of course, there are nowadays the new approach. Instead of focusing only on the virus, now there is this urgent need to focus on the host also, all the host mechanisms that are hijacked. And so there have been some proteomic approaches that are giving us some guidelines as to how to discover drugs towards treating especially these multi-organ damage problems. Okay. Then finally, as I said, vaccines seem to be the vital to handle this pandemic, to fight this pandemic really. And of course, the drugs development will take place, but it is going to be a longer, it's not going to happen overnight. But there seems to be this vaccine research is going, as you know, at a warp speed, and there is hope that we will have some vaccines by the end of this year or early part of next year. So with that said, remember these human coronaviruses are not necessarily new. We have these come from the Nidovirales family. They show subgenomic mRNAs, by the way. And these human coronaviruses have come into our lives long time ago in the form of two lineages, alpha and beta lineages. These so-called common coronaviruses, they cause common cold. They are of low pathogenicity. And this 229E and L63, they are alpha coronaviruses. They basically induce common cold on a seasonal basis. And then we have these two beta coronaviruses. They also induce common cold, OC43 and HKU1. Okay, but what we are now seeing is these beta coronaviruses, SARS-CoV, MERS-CoV, and SARS-CoV-2 since 2002. And we believe that the SARS-CoV got into humans via civet cats. And basically, these are all bat coronaviruses. Obviously, there's not much of human interaction with bats. So there has to be an intermediate. And so the logic goes that based on all the virus phylogeny studies, we believe that it is SARS-CoV from bats got into cats, and through natural selection somehow emerged in humans. Okay. Similarly, MERS-CoV, the intermediate is camel, and SARS-CoV-2, we believe through phylogeny studies that somehow they have emerged in humans via pangolins. Okay, we don't have time to go through all this, but the key thing to note is all these coronaviruses. By the way, SARS-CoV-2, for whatever reason, it is not in circulation. Either it is mutated out or not able to, there is no significant reservoir. So SARS-CoV-2 has not been in circulation, believe it or not, since 2005. Hope this can happen to SARS-CoV-2 soon. And so SARS-CoV persisted on Earth for 3 years, I should say. At least we have not seen it since 2005. Now, MERS-CoV, on the other hand, is still in circulation, but sporadically. And this is has a very small or not, but highly, it can induce high fatality, because it is the fatality rate can be up to 35% compared to what SARS-CoV-2 is doing, which is minuscule at the moment. Okay, bottom line is that these are all coronaviruses belonging to beta coronavirus family, and they all cause severe pathogenicity, what we call as SARS. And now, having said that, SARS-CoV-2, which came in 2002, an enormous amount of science has been done. Basically, we are jump starting based on the science that we know of SARS-CoV and MERS-CoV. Okay. Now let's speak of the spike protein. First, that means we need to understand the structure of SARS-CoV-2. SARS-CoV-2 is an enveloped virus. Okay, it's very important to realize that. And it is a single-stranded positive sense RNA, RNA virus. Okay, so single-stranded positive sense RNA virus, which is a very big virus. It's 30 kilobases long, and it has recently, this couple of days ago, there was a Nature paper which claims that there are more than 23 open reading frames in SARS-CoV-2, which is very big. As we 2-3 months ago, we were just thinking of around something like 16, 17. Now they say there are more than 20 open reading frames. That means we don't know many proteins that are out in this that are encoded in this virus. Okay, and there are 4 structural proteins. This is well understood. And in the 2 open reading frames that we'll talk about, there are 16 non-structural proteins. NSP stands for non-structural proteins. There are 9 accessory proteins. Okay, and this is the typical schematic that we write for the virus. And this is the envelope that is stolen from the endoplasmic reticulum, let's say. And this is the membrane protein. And then there is this nucleoprotein that covers the RNA and forms what we call nucleocapsid. That's a protective of the RNA. And then the envelope protein. All these have multiplexed functions. And this is the spike protein. This is the one that enables the virus to interact with many target cells in the human body. Okay, the spike protein actually, as you can see, is a trimer of so-called S protein. There are 3 S proteins, so together form this spike protein. The spike proteins are 10 nanometers in length. There are 300 copies per virion particle on the rough estimates. Okay, and these are published in this journal, in this article. And these S proteins are roughly around 1300 amino acids long. And this S subprotein again has 2 subunits, S1 and S2. You can almost make out by the structure that is written here, shown here. S1 is so-called the globular domain. This S2 is the stop domain. Okay, the stop domain. Okay, this globular domain has the receptor binding domain for ACE2. Remember, ACE2 is the receptor on the target cells in the human body, with which this globular domain interacts to gain entry into the cells, right? And this is the stop domain. All right. Now, the stop domain also has what we call fusion protein. And this fusion protein actually inserts into the host cell and after the cleavage of this S1 so-called globular domain. Okay, with that said, let's take say a few things about other structural proteins. I want to focus, because there are too many details here, I want to focus on one. Let's say talk about N protein and M protein. See, these proteins do multiple jobs. Okay, yes, they are decorated here on the membrane. What does it do? This is a central organizer of viral assembly. We don't have time to discuss in detail, but it's very important to realize that these forms are important in the viral assembly. Oops, sorry. And then it also interacts with many host proteins. That's the main point that I want to remember. And similarly, when you look at nucleoprotein here, yes, this binds to RNA, as I said. It interacts with many host proteins. Okay, and similarly, E protein interacts with many. It induces stress response in the host cells. Okay, very important to note these details. And this virus, as we stand, as we notice today, it is pretty stable. It is showing roughly 2 mutations per month. As most of you may have noted, there is one mutation that has come to prominence, that is T614G mutation in the S protein. Okay, and this mutation was discussed at length in a paper in Cell in the last couple of weeks ago. And what they claim is that this mutation has transitioned to the whole world, more or less nearly 100% is covered with this particular mutation. And that means all the new viruses, all the people who are getting infected now possess this mutation. That is, instead of aspartic acid in the Wuhan virus at the 614th location, now it is glycine. And this particular article claimed that this can cause increased, they said you can have, they find that these patients have increased viral loads. Okay, this is being debated, but one thing that we can say that this mutation has led to formation of a new clade called G clade, but not necessarily highly pathogenic. That's the claim by many scientists. Of course, the publications are going to come out very soon based on the studies. But at the moment, we can say that, of course, the science is changing, as you know, every day. But at the moment, what people believe is that this mutation is not necessarily causing increased disease severity. Okay. So bottom line is the significance of this is that the virus is stable. Supposing we have a vaccine down the road and that vaccine need not be changed periodically. That's the take-home point. And now let's see how the virus enters. Okay, I told you already the spike protein enables viral entry. We already, Dr. Madhyastha told us that ACE2 is the receptor on the host cells. Okay, now this this S protein binds to ACE2, and at the same time, there has to be some kind of protein cleavage of the S protein, and then the virus gains entry into the cell. Okay, so but remember, this ACE2 is angiotensin converting enzyme. It is actually a carboxypeptidase, but the catalytic activity of ACE2 is not important. Okay, what is important is for the cleavage of the spike protein that enables the fusion of this virus with the membrane, you need this enzyme, TMPRSS2. It's a serine protease, TMPRSS2, and this can cleave the S protein, specifically in particular one domain, which allows these membranes to virus to fuse with the plasma membrane of the host cell and gain entry. Okay. By the way, please note that ACE2 is upregulated in various tissues during COVID-19. Now I want to tell you a lot more about spike protein, but we have limited time. But because in the science, knowledge about spike protein is very critical for vaccine development and also for understanding how the viral entry occurs. But key thing is, as I said, spike protein is basically a trimer of S protein, and it has 2 domains. And this cleavage of S1 and S2 is the first step for viral entry. Either this has to occur even before the virus binds to the host cell, it can occur, or it can occur after binding to the host cell. So in any case, what we have is these S proteins possess 2 cleavage sites. Okay, what is called a furin cleavage site and then what is called an S2 prime cleavage site. This cleavage site is sort of novel to coronavirus 2. And this, they believe, is responsible for high affinity of this coronavirus 2 to ACE2 receptor. And that is probably one of the reasons which might be driving this contagiousness. And now they believe that this furin cleavage site cleavage can occur before budding of the virus. Please remember this. But this S2 prime cleavage site is broken down by this enzyme, TMPRSS2. So that means AS2 and TMPRSS2 has to be co-expressed. They have to be co-expressed for the S2 prime cleavage. Okay, then only you can have a fusion of the virus with the target cell. Okay, and I'll skip this detail. And now here is how it looks. By the way, there are three, as you know, this is the S domain now. This is the S2 domain, and this is the stalk portion of the virus, of the spike protein. This is the globular domain. This is somewhere here. You have the so-called binding site to ACE2 receptors. Okay, and the first thing that has to happen is the cleavage between S1 and S2. This is the this cleavage site. This is called the furin cleavage site. This is very novel to coronavirus 2, that is SARS-CoV-2. And then this cleavage may occur as the virus comes out of the host cells. That means after multiplication. Okay, this TMPRSS2 cleavage site, this is S2 prime cleavage site, is accomplished by TMPRSS2 enzyme, which is co-expressed along with ACE2 in many target cells. So bottom line now, let's see how ACE2 is expressed. So ACE2 is expressed widely in the body, especially in tremendous densities in enterocytes, kidney, and cardiovascular system. But definitely it is present in type 1 and type 2 alveolar epithelial cells, and that means in the lungs, in pneumocytes, and also in the upper airway, along the airway passage, and even in the nasal cavity and in the oral mucosa. Okay, so bottom line is, remember, type 2 pneumocytes possess ACE2 receptors. And now this ACE2, as we just heard, it does many things, by the way, in terms of the physiological functions in our body. And all of you know this, angiotensinogen is converted to angiotensin 1 by renin. This renin can be converted to this vasodilatory agent, angiotensin 1,9 by ACE2. That is one of its functions. And at the same time, remember, this ACE, which is typically we inhibit by ACE inhibitors for treating hypertension, this converts angiotensin 1 to angiotensin 2. And angiotensin 2, as you know, is the most potent vasoconstrictor in the body. And this not only does vasoconstriction, this also drives aldosterone release from the zona glomerulosa cells. And that aldosterone, of course, induces sodium and water retention and increases blood volume. Now, what happens is, this angiotensin 2 is also converted by ACE2 to angiotensin 1,7. So basically, this ACE2 has a negative impact on renin-angiotensin axis. Please remember this. So ACE2 has a negative impact on renin-angiotensin axis. So this is very, very critical to all our discussion, because we need to understand, to really focus in on pharmacology, we need to know this renin-angiotensin axis. And by the way, this ACE2 is also very homologous to ACE, and it's also a metallocarboxypeptidase. That means it's zinc-based. And ACE2 is not inhibited by ACE inhibitors. Please remember this. And then also it's found always in the apical membrane. And as I told you early on, ACE2 catalytic activity is not necessary for viral entry. Now let's see the virus life cycle. Very simple. So this is the genome of the virus. It's pretty long. And what happens initially is, remember, this is a positive sense single-stranded RNA. That means it acts like an mRNA once it gets into the cell. So it gets translated the moment it gets into the cell and undergoes disassembly. Okay. So what happens is, in this virus, these two so-called open reading frames are translated. They undergo what we call primary translation. And so these two, and you get polyproteins. And the way it works is, the two kinds of polyproteins are produced. One is this PP1A, and another one is PP1AB. It's a long one. And this is encoded by these two open reading frames. And this is accomplished by ribosomal frame shifting, which is a mechanism that we don't have to deal with. right now. So these two polyproteins are expressed the moment by translation, the moment virus gets into the cell. And then they undergo autoproteolysis. And look at this, there is one enzyme here, and that gets released by autoproteolysis. This is also gets released, again, autocleaved from this polyprotein. And these undergo and cleave these polypeptides and release all these molecules. This particular protein called exonuclease and helicase, and then RNA-dependent RNA polymerase, which we call RDRP. And these sort of co-assemble in what we call double membrane vesicles, double membrane vesicles. And this is where RNA replication takes place for the virus. Okay, let me show you that. So here is the coronavirus is binding to ACE2. Of course, the cleavage of S2 domain occurs by TMPRSS2. And then you have the virus enters and undergoes disassembly automatically. The way the virus enters, it comes disassembled, and the RNA undergoes directly translation, and you get this polyprotein produced. And these two polyproteins, they undergo autoproteolysis, and you get all these proteins. This is RDRP, helicase, and exonuclease. Okay. So the moment these automatically get assembled in the so-called double membrane nucleus as vesicles, and there is an interesting type of transcription that occurs called discontinuous transcription, through which you get this whole genome copied as into positive strands, of course. You will have negative strands also formed, and then positive strands are formed. And then you also have subgenomic transcription, which are mRNAs for all these structural proteins and other accessory proteins. So, and some of them have to undergo translation with ribosomes attached to the endoplasmic reticulum, especially, as I told you, this S protein is highly glycosylated, specifically N glycosylation. That means it has to occur in the endoplasmic reticulum. And this N protein, it does not require any of that. And also it simply binds to this RNA. And then the vesicles that are formed out of this endoplasmic reticulum, the viruses bud off with proper assembly, self-assembly. And somewhere before budding, remember I told you about that S1, S2 cleavage, furin cleavage site, that cleavage may occur. And so this is now a ready virus that can go and infect the neighboring cells. So this is how it works. So the key things to remember is discontinuous transcription, then there is this autoproteolysis and subgenomic mRNA formation, etc. Okay, and many of these steps are targets of, for example, these polyproteins that are, and this RDRP, these are all pharmacological targets for blocking viral multiplication. Now let's think about how the virus is transmitted very briefly, and mainly by respiratory droplets. There is not much scope for fecal, oral, or blood-borne transmission, and aerosol transmission is likely, but not very important, I think. But in any case, that's something to be you need to be cautious about. And interestingly, vertical transmission in pregnancy is also not reported, and virus is not present in the breast milk. These are some important points that I thought would be important to all the listeners. And then let's think about the clinical features of COVID-19. Okay, so all of you know this, and especially when once are infected, there is an incubation period, roughly 5.2 days on the median, and it can vary from 2 to 14 days. And the symptoms manifest generally when the person becomes symptomatic, an infected person becomes symptomatic. Typically, there is fever to a large extent. Most patients show fever and dry cough, and then difficulty breathing. Okay, when the breathing difficulty sets in, most people end up going to the hospital, and after hospital admission, and they develop dyspnea, that is shortness of breath, and very quickly they may end up, if many people recover, but many people end up with ARDS, and a few people may end up even ICU because of multi-organ damage or severe other complications. Basically, in these people, they should have some comorbidities or severely old people. Okay, advanced age. Okay, so these are the risk factors. Okay, advanced age and diabetes mellitus, cardiovascular disease, is part of that. And when I say here cardiovascular, probably it refers to the heart, and then chronic lung disease, obesity, etc., etc. Okay, these are all well-known risk factors. So what is the immune response? Remember, we are trying to understand, I told you early on, that you have asymptomatic phase, and then you can end up with a situation where there is dyspnea, and then eventually end up having a phase for hyperinflammatory syndrome where multiple organs are affected. And that means in all of these, cytokines are implicated. So how do we get these cytokines? So just a quick review of this. And we can spend a lot of time on this, but we don't have a lot of that kind of time right today, right? So what we do, what I want to say is that some of the virus particles, as they undergo transcription during the intermediate RNA, can form what we call pathogen-associated molecular patterns. For example, the ssRNA in some phase can activate TLR receptor, TLR7 and 8, and also when this ssRNA gets associated with another genome, can form double-stranded RNA and get recognized by these RIG-I and MDF5 sensors. And these downstream of signaling of these double-stranded RNA sensors, you end up getting activation of these transcription factors. These are interferon regulatory factors, okay? And they drive production of interferons, okay? And at the same time, these TLRs here, downstream of this through MYD88 signaling, they can drive the activation of NF-kappa B transcription factor, which can induce many cytokines like IL-6 and TNF-alpha. These are hallmarks of COVID-19, by the way. And eventually, this virus itself can get recognized by the macrophages or antigen-presenting cells, and they communicate the knowledge to naive T cells, and you end up forming CD4 positive T cells and CD8 positive T cells. These also put out lots of cytokines, and if they're not, this whole phenomena is not controlled, you get hyperinflammation. Okay, and this is we are showing with respect to lung. This can occur in any organ because, as I told you, ACE2 is expressed in many organs. Okay, and by the way, typically out of even the B cell activation leads to antibodies that are nucleoprotein specific or S protein specific. So this is what you would see in a person after vaccination as well. Now let us see what are the complications that you typically see. You notice always, people die of ARDS. So this is attributed to cytokine storm. So instead of discussing the mechanism, I want to tell you what happens here. So basically, you have this is the alveolus, right? And this air space gets flooded with because the endothelial barrier is broken here, and even the epithelial barrier is broken. Okay, and this, by the way, the type 2 pneumocytes. And there is, you can see the human blood rushing into the alveolus. There is thrombosis here, and there is a lot of inflammatory cells, and there is fibrosis. And so you can imagine everything that can go wrong in the lungs has happened in the alveolus, has happened here, with significant inflammation, a lot of cytokines, and damaged cells, and even the red blood cells have appeared in the alveolus, and neutrophils and macrophages. And so basically, ARDS refers to a severe situation of inflammation where the alveolar, you have pulmonary edema, and the person oxygen exchange falls, becomes very small, and the person dies. What are the other complications that you see secondary to respiratory failure? Shortly after the ARDS manifests shortly after dyspnea. Then later on, if the situation is not controlled, you will see inflammatory complications. At a laboratory level, you see formation of D dimers, ferritin, increase in SCRP, and increase in the levels of pro-inflammatory cytokines, and abnormal coagulation tests and lymphopenia, etc. And at the same time, you will notice cardiac injury, arrhythmia, and whatnot. You can see cardiac injury in the lab test. You will see it as elevated HS, high sensitivity troponin, or creatinine kinase MB, and oftentimes you notice shock, septic shock. Okay, that is one of the reasons you see certain symptoms in the central nervous system as well. And many times, as you may have seen, a reporting of these thromboembolic complications involving acute stroke, for example. Generally, what is not known is secondary infections. So you get all other complications. So we need to treat these. Do you need to treat viral multiplication during this stage of the complications? Generally not. So you can choose dexamethasone when you are in these kinds of severe complications, because the viral multiplication is not very important in this phase, hyperinflammatory phase of the COVID-19. So now let us think about one thing, what may be underlying septic shock very quickly. So there is one interesting study that came about in the last 2 weeks, and that is by these authors. And what they did was they looked at one old study, basically an RNA-seq study, and in which that obtained the samples from bronchoalveolar lavage fluid, okay, from 9 patients. And they also took some 40 patients control data from some other studies reported in the literature, and they found differential gene expression along the renin-angiotensin axis as well as bradykinin pathways. What they found was, in a nutshell, simply said, they found ACE2 expression to be reduced, ACE expression to be reduced, ACE2 expression to be upregulated. And they found changes in the kallikrein-kinin system predictive of a bradykinin storm. Okay, I'll show you what it does. And all of you know this bradykinin, what it does. It can induce pain, it can activate nociceptors, and it can cause vasodilation of arterioles and constriction of venules, such that and also at the same time breakdown of endothelial tight junctions, and such that you can have fluid loss across the capillaries and causing edema. And remember, sometimes bradykinin is associated with angioedema, which is another disease by itself. Okay, so bottom line is bradykinin storm can occur according to this RNA-seq analysis. So they also found gene expression suggestive of overaccumulation of hyaluronic acid. That's very interesting, because that is also noted in the lungs in the autopsy studies. So let me tell you what happens. This is an axis. This is a renin-angiotensin axis that all of you are familiar with. And I just want to highlight some key data here. And remember, this is angiotensinogen here. And renin gets you angiotensin 1, which is cleaved to angiotensin 2 by ACE. And what they found was renin was upregulated in these patients by 380-fold. And ACE was downregulated by 8 times. And remember, I told you angiotensin 2 is converted to angiotensin 1,7. This ACE2 is upregulated in COVID-19. Okay, so at the same time, I told you that ACE2, because of upregulation, it also converts angiotensin 1 to angiotensin 1,9 that is one step. And this angiotensin 1,9 acts on AT2 receptors. Okay, so this is upregulated by 177-fold. So what is happening is in COVID-19, based on just this RNA-seq data, we can tell you that there is significant drive towards vasodilation and natriuresis. That means volume loss as well as vasodilation. Something predictive of septic shock. Okay, predictive of septic shock. Even though the AT1 is upregulated by 431 times, but you're not getting enough ligand for this receptor. And so these people who studied, who conducted this study, they predict that there is significant vasodilation and natriuresis because of AT2 in the COVID-19 disease. This is all based on RNA-seq data from the BAL fluid, from the BAL samples. And look at this bradykinin system, what happens. And this is just we discussed just now, right? And now in the bradykinin system, it's complicated, but I can tell you very simple. So this bradykinin is a vasodilator to start with. Okay, it is formed from plasma kallikrein or is formed from tissue kallikreins. Okay, and the tissue kallikrein produces this kallidin, which is analogous to bradykinin. In fact, kallidin can be converted to bradykinin. And these two agents, bradykinin and kallidin, can interact with B2 receptors. At the same time, this bradykinin gets converted into this active metabolite by this kinase 1, which was found to be upregulated by 671 times. Okay, very profound. At the same time, look at this receptor. This active metabolites, this bradykinin and this kallidin, can interact with this B1 receptor, which is highly upregulated. This is typically not a constitutive receptor. It's a bradykinin receptor, not a constitutive receptor. This is a constitutive receptor. And this is present or is expressed now in COVID-19 patients based on the RNA-seq data, and by 3,000-fold upregulated. And as is known, it's typically upregulated during inflammation. So what is downstream of B1 receptor activation? You basically get release of vasodilation, you get pain, you get leukocyte recruitment, basically inflammation, and breakdown of vascular barrier and dry cough. So all symptoms that you are thinking to explain based on some physiological information, you have it here. Basically, you have upregulation of these B1 receptors and downregulation of angiotensin II activity. And that's what we see in COVID-19. So there is all of a sudden a drive towards development of B1 receptor antagonists, okay? And also to see what other pathways can be, but this is the main pathway. And also this enzyme, there is one enzyme that controls the levels of plasma kallikrein, and even that is downregulated, because of which this is upregulated. So what they found is that I just told you, this ACE, renin, and ACE and angiotensinogen, they're all, the mechanisms are shifted towards vasodilation and natriuresis. So this may, they predict that this may underlie the septic shock that we see in this advanced COVID-19 patients. And at the same time, these bradykinin receptors are severely upregulated, such that you get this edema, pulmonary edema, and severe shock. And finally, I want to say just here that the hyaluronic acid accumulation takes place in the alveolar space. You have a jello in the alveoli, and obviously the gas exchange becomes difficult, and so dyspnea manifests very readily. So with all that said, now we can think about pharmacological targets. Okay, so we have a rough idea about the immune response. We have a general idea about what could be underlying the what is the general mechanisms and involving associated with virus life cycle, and so we can think about some of the drugs. So let's talk about targeting the virus directly. Obviously, very simple. We can think about viral entry inhibitors and viral proteases, because these are specific. The human proteases cannot break down the polyproteins of the virus, so these proteases can be inhibited to curb viral multiplication. And similarly, this viral RDRP, the RNA-dependent RNA polymerase. And so bottom line is that these kinds of drugs to block the viral multiplication, you can use it in the viral multiplication phase, not during not effective in the later stages, especially during a hyperinflammation stage. That is typically the thought at the moment. And now these drugs to overcome immunopathology. Okay, this is what happens during you have severe immunopathology during hyperinflammation. So then you can go for drugs to target the cytokines, and drugs to bring about immunosuppression, such as dexamethasone, and basically immunomodulators. And these should not be used during this early stage. Otherwise, you will end up enhancing viral multiplication. And then we can think about the viral target host mechanism. Okay, we can think about the specific mechanisms in the host that can be altered to combat the disease process. And of course, you need supportive treatment depending on the patient as needed. Now, I'm going to report negative data. Obviously, there are, but what the idea is that we know that what can be targeted, and there is some small amount of efficacy. We have to tweak these drugs. These were developed in the context of SARS-CoV and MERS-CoV. Now we have to use these agents, and these are being, basically they have to be improvised, so that we see some efficacy against SARS-CoV-2. But what we know is these are prototypical TMPRSS2 inhibitors, nefamostat and chemostat, and this is Orbidol or umefenibart. Okay, so these have specific mechanisms. Instead of focusing on the mechanisms, what I want to say is that right now all the clinical trials have not shown tremendous efficacy of these drugs. Okay, but still the clinical trials are ongoing right now. Okay, and these are called fusion inhibitors. They simply, these two especially prevent TMPRSS2, and this prevents the fusion of the viral protein, that is the spike protein, by preventing trimerization of the S protein. Okay, and also it affects the membrane, the viral envelope as well. And now this lopinavir, all of you know about lopinavir. It is a standard protease inhibitor for treatment of HIV. It's usually administered along with ritonavir. And ritonavir basically inhibits P450 and jacks up the half-life of, half-life of lopinavir, by the way. And that's how we use it. And then what is thought is that this lopinavir binds to this main protease. That is, remember I told you this the virus after disassembly, the first step that occurs is these proteases are formed, and these proteases bring about the proteolysis of these polyproteins, right, that are produced by this RNA virus. And now this lopinavir is thought to bind to this main protease. Okay, there are two proteases I told you, and the main protein is also called as 3C-like protease. And this lopinavir seems to have affinity for this particular protein. Okay, and but actual treatment, when you go and give lopinavir along with the ritonavir, the efficacy is not there. Okay, so obviously we need to fine tune. But there is one important fact that you need to remember. HIV protease is an aspartyl protease. But what we are seeing in this MPRO is actually a thiol protease. There's a cysteine protease, basically. So how can we expect this lopinavir to work on this MPRO, which is a cysteine protease? Anyway, alternatives are also being discussed, but overall, we can develop, based on the knowledge of lopinavir and other protease inhibitors, we can definitely develop down the road these inhibitors for MPRO. And that is in fact going on right now. And now remdesivir, all of you have heard about this, and this showed modest efficacy, I should say very little efficacy in the clinical trials. And that too, some people have questioned that the clinical efficacy. Basically, this forms a fast building block. Okay, it substitutes for adenine nucleotide. It's actually a pro drug. It's converted to nucleotide form once it is administered, once it gets into the host cell, and then this basically induces what's called delayed chain termination. Okay, and thereby blocks transcription. So typically we expect this drug RDRP to be inhibited, and inhibition of transcription to take place, and thereby block the viral transcription. But the efficacy has been very, very little. But we know that remdesivir is very good against Ebola, at least in vitro. And similarly, remdesivir is very good against SARS-CoV in vitro, but we don't understand why it is not working when it is injected. By the way, the drug is right now being injected by IV infusion over 30 to 120 minutes. It has a very short half-life, but this is the strategy right now. And there are studies where this remdesivir is trying to be administered by inhalation, but I don't think I've seen those results. But we have to say it has pretty significant binding affinity with MPRO, and hopefully this drug we can sort out and improve the efficacy of remdesivir or its analogs down the road. And there is this another interesting drug. This is called favipiravir. This is used in Japan for severe influenza. So it's modest side effects. So it can be sort of, if it works out for SARS-CoV-2, it would have been great. What it does is it brings about promiscuous base pairing, and thereby induces lethal mutagenesis. And that's how it makes the RDRP to stop its work function. That means stop the transcription activity. By the way, this is also a prodrug. This enzyme converts it into nucleotide form, and it can be given orally as well as intravenously. So far, this drug has not shown efficacy to the best of my knowledge, but still the clinical trials are ongoing. And these two, I don't want to say anything much other than one fact that I can say zinc is an inhibitor of tissue kallikrein. That's something I noticed just now. At the same time, hydroxychloroquine is thought to be an ionophore for zinc, if that is of usefulness in immunomodulation, and that could be an advantage. But overall, in this country, we generally question about the effectiveness of HCQ, and also the toxicity is highly debated. Somehow, this drug has not found a place for a treatment of COVID-19 so far. And this N-acetylcysteine is being used on an ad hoc basis. We don't understand its efficacy or rationale for use very well, but nonetheless, we all know that it is an antioxidant. And now let's think about the cytokines, how to handle the cytokines. And if you do those lab studies, we know that we have plenty of IL-6 in the circulation in COVID-19 and during hyperinflammatory phase. So initially, this expensive antibody, this is a humanized monoclonal antibody against IL-6 receptor. It's called tocilizumab, and this was deployed. And I think the clinical trials have not shown significant efficacy, but I think is still in many clinics they use this drug. It's very expensive. It's administered by IV infusion over an hour, and it does have some side effects, especially during pregnancy. Of course, that's one thing that needs to be watched. People have to think. But what it does is basically by binding to this IL-6 receptor, it prevents the signal transduction through this GP130. Remember this, this is the IL-6 receptor. Let's say this is the IL-6 receptor. So this is the IL-6, this is the IL-6 receptor. And when IL-6, this is IL-6, when IL-6 binds, the signal transduction occurs through this GP130, and it drives the JAK-STAT signaling. And what happens is this tocilizumab binds to the receptor and prevents interleukin from binding here. Okay, so basically tocilizumab and saralimumab both are antibodies against monoclonal antibodies. They block, they bind to the IL-6 receptor, be it in the soluble form or insoluble membrane-bound form of the receptor, and prevent interleukin-6 from binding to it. Yes, it's effective in many other diseases where it has been approved before, but so far efficacy against COVID-19 has been marginal or not been properly disclosed. So then we have, obviously we just saw the cytokines, of course, the downstream of the cytokine receptors, you have JAK-STAT pathway orchestrated. That means that is what is mobilized, leading to additional transcription of cytokines. So we have a small molecule inhibitors for JAK-STAT pathway, and you have this drug which is used for inhibits both JAK1 and 2, and then baricitinib is also used. These are all approved for rheumatoid arthritis, and this also inhibits JAK1 and 2. And these have been used but not found efficacy. It means at least the clinical trials have not been properly finalized. The data have not been finalized. And if supposing there had been a significant efficacy, they would have announced it by now. And then similarly, we have IL-1 antagonists and many TNF-alpha inhibitors in the form of, for example, Humira, which is a very popular drug against, for example, Crohn's disease, and infliximab, again, very popular against Crohn's disease or ulcerative colitis and whatnot. So, but again, these agents are available, being tested, being used, but not, the efficacy has not been demonstrated. Then this is a drug, dexamethasone is a vanilla agent, is a glucocorticoid, which is both an anti-inflammatory and immunosuppressive. We have to be very careful to use this only in severe COVID-19 patients. So right now, the idea is to use this, not use for treatment in patients who do not require supplemental oxygen. That means who are not reached severe or advanced COVID-19 situation. Okay. So anyway, this is given by oral route, but inhalational, for example, the budesonide, which is used popularly for asthma, which is being this one is also prescribed for asthma. These drugs are being tested for treating COVID-19. Very quickly, I want to tell you about there are, I hope I have time, and let me tell you a few things about the proteomic approaches being developed to target host mechanisms. So one study that came about just a few weeks ago, very, very interesting study. What they did was, they did the proteomics. So they expressed several proteins of the SARS-CoV-2 in HEC293 cells, okay? And they did the mass spectroscopic analysis to do protein-protein interaction studies, okay? So they found that this SARS-CoV-2 protein interacts with 332 proteins of the human body. Okay, then they went about and studied where these proteins are found in the human body. they found it is expressed in various parts, especially in the lungs. Okay, so this is a differential protein expression of the proteins that interact with SARS-CoV-2. Okay, that means SARS-CoV-2 proteins interact with many host proteins. Obviously, they're doing something to them, so to elicit the disease, right? So what they did was they found, for example, nucleoproteins of SARS-CoV-2. Okay, that is the N protein. They showed that it can interact with these components of so-called casein kinase. Okay, so these are important apparently in some stress signaling. And they found this M protein to be associated with proteins associated with ER morphology and mitochondrial metabolism, solute transport. Remember, there are 26 other proteins. They showed this kind of diagrams for all the 26 proteins in this journal. I think it's a Nature journal. Okay, and then they went one step further. This came out last week or so, where they did the phosphorylation assay of the proteins. So they did mass spectrometric analysis of phosphoproteomics. They basically infected this Vero E6 cell. These are green monkey cells. They possess ACE2 and TMPRSS2, etc. So they're highly infected, easily infected by SARS-CoV-2. And of course, these are monkey cells, remember. And what they did was they looked at the protein-protein interactions at the phosphoproteomics level. They found that there was roughly 40 proteins were showed prominent differential phosphorylation. And out of that, these 14 proteins, these proteins are susceptible to, rather, the phosphorylation could be affected by various proteomic studies. They found 49 kinases can bring about changes in the phosphorylation of these 40 proteins. And one of them was CK2. Okay, and we have shown in the previous slide, right, that this N protein interacts with these two genes. And this gene, for example, or this protein rather, is a regulatory subunit of casein kinase, and this is a catalytic subunit of casein kinase, and these interact with N protein. So what they found is that this casein kinase interacts with N protein and drive various signaling mechanisms which affect the cytoskeletal organization. They found that activation of the CK2 led to filopodial protrusions in these Vero 6 cells, and that was responsible for facilitating virus to bud out from this virus, from this E6 cells. So now you know a mechanism which is responsible for viral budding. That means if you can inhibit this, it's going to be very efficacious, right? So indeed, they tried to inhibit this cytokine, this casein kinase, and that led to decrease in viral activity. That means viral proliferation. So what they've done is these are big mega scale techniques through which we have now found a way to address the host proteins that are affected by the virus. Now let me go to the vaccines. Of course, the last hope, right? We have seen all the drugs, whatever be it remdesivir or convalescent serum that I put the slides, dexamethasone or IL-6 antagonist, they all have marginal efficacy, of course, with appropriate supportive care. And still the fatality rates in hospitalized patients is pretty high. And we are all scared of this fatality or even undergoing this hyperinflammatory phase, because of which the whole economy has come to a halt, right? All the activities of human life has been paralyzed. So we need some big thing, okay? Big urgent approach is needed. And that's where this vaccination comes into picture. You need vaccination to bring down R0 and to induce herd immunity, as Dr. Bhandari was telling us, okay? You want to break the chain of transmission. Of course, the vaccines that we are developing may not be efficacious in everyone, and not everyone may be able to participate in vaccination. We are pregnant women or whatnot. So, but what is important is only 70% of the patients of this the population, if they show immunity towards this vaccine, towards this infectious agent, then we would have induced herd immunity enough to break the circulation. Okay, so many vaccines have been developed. Many of them are so-called plug and play like vaccines. They're suitable for rapid manufacturing and also to change them very quickly if supposing the virus shows some mutations, enormous mutations. And looks like based on the phase 2 clinical trials so far, many of the vaccines are already in phase 3 clinical trials. And success in phase 3 leads to release of the vaccines to general public. And now phase 2 clinical studies have shown that we need booster shots as well. That means, remember, there are 7 billion people or so in the world. We need an enormous amount of vaccine shots, right? But one thing is, this development is taking place at warp speed. To be honest, everybody is working around the clock, and there is enormous amount of collaboration and whatnot. Things are working. Let me touch upon two vaccines that sort of are in a few vaccines that are in phase 3 clinical trials. And this is by Moderna. This is an mRNA vaccine. The mRNA is for S protein. It's put in a lipid nanoparticle and injected. Okay. And being a lipid nanoparticle, it goes into the cells at the site of injection. And of course, you get these proteins formed, which are highly antigenic. And you get the antigen presenting cells taking it to the lymph node and producing the immune response. Okay. And then you have, this has been, by the way, this is already in phase 3 clinical trials. Phase 2 studies were very good, and very good within quotes. Of course, we wish we had more data, but whatever data they have put out seems to be very good. And now this is another mRNA virus. Here, multiple messenger RNAs are put here in the lipid nanoparticle, again injected, the same strategy. But the idea is there are multiple messenger RNAs. Okay, so this may be more efficacious depending on how you look at it. Okay, and of course, the one that is from China, this one from China, this is, they are using viral vectors to get the S protein into the human body, and in the form of vaccine. So this is the adenoviral vector into which they have put in this a gene encapsulating this S protein or encoding the S protein. And this again, these viral vectors, the advantage is they show profound immune response. And so they think that sometimes you may not need an adjuvant when you inject this. And so you get plenty of these proteins produced and you get the immune response. Again, they are already in phase 3 clinical trials. They're very aggressive, and the studies are ongoing. And soon I think we'll have more data coming out in the next 2-3 months from all companies that I'm talking about. And this is the, this is Oxford adenoviral vector. This is a very complicated adenoviral vector, but again, they're using S protein as the antigen and being targeted. Again, I just want to tell you that they are in phase 3 clinical trials. So I want to summarize and allow some questions to come from all of you, and allow some time for questions. And basically what we have is there's no magic bullet. Okay, there's no magic bullet. And we know that there are 3 major phases of the disease, so-called the asymptomatic phase, during which people are able to infect others, but unfortunately they're asymptomatic. We don't know, they will not have gone through the test, so they are mixing with the general public and causing infection. Okay, so it's highly contagious because of that. And in the bottom line is there's no magic bullet right now, pharmacological drug, there's nothing like that. So we need vaccine development. This is of paramount importance. So everybody has to work towards this. And we have to be aware that these billions of doses are required, and logistics of administration becomes a challenge. And everybody has to gear up right now. We have to foresee the challenges and sort of come together and come up with strategies for administration at large scale, and convincing people at the same time having the logistics also already ready. And then we have to think about the pharmacological agents. So far, the targets are self-evident. Okay, we know the targets to handle the viral multiplication. So far we have repurposed drugs against RDRP, proteases, and viral entries. We need to come up with derivatives of this based on so-called bioinformatics studies, and hopefully have increase their efficacy. At the same time, we have to come up with a host inflammatory response, especially in the context of vasculature, heart, kidney, and the lungs itself. And as somebody early on said, what we need is enormous amount of collaborative, interdisciplinary, and large-scale operations. And this is not, we cannot win it just by ourselves, just a small few people. And we need a large number of people to get together to bring about this massive, to handle this massive challenge. Okay, I thank you very much. I don't know if I took a long time, but I appreciate everything. And so please feel free to ask me some questions.
So, hello, Shilpa. Can I take out?
Yes, please.
Yes, sir.
Yeah.
Yes, sir. You can take up.
Yeah. So excellent to speak, Professor Srinivas. It's a really very good, very inspirational and motivational and challenge-oriented thoughts. Especially in the last sentence, that collaboration, of course, is required for the every challenge you mentioned in that one. Thank you very much.
Okay,
There are several questions from around 300 participants.
Yeah. So let's say one thing. You can always, you have my email address. I'll make my slides available to Dr. Sucheta Kumari and to you.
Yeah.
And they can send me. I would love to interact with all the people if possible, and basically get lots of questions and also interact. Do research collaborations, etc. But we can answer some questions very quickly.
Yeah, because one Dr. Purnima from she said there is a role of lot of inflammatory pathways being activated. Is there any drug repurposing tied with the drugs targeting pain ion channels?
Okay. Yes. Specifically, very interesting you asked me that question, because just I came across a study where sigma-1 receptors are thought to be involved. And sigma-1 receptors, they are the opioid receptors are related to opioid family receptors. And yes, that can be tested. I don't think anything, to the best of my knowledge, no other studies. And I would like to ask Dr. Purnima to send me an email, and I will tell her about the sigma-1 receptors. Basically, you need to block the sigma-1 receptors in this case, not actually activate. And that is one thing that I have come across. But otherwise, of course, now that I've told you about bradykinin, let's think about bradykinin antagonists. Okay, B1 antagonists, what we need. And by the way, there is a table that I can send about B1 antagonists that we can test. First of all, we need to understand expression profile of B1 receptors in various parts, and then we can see how we can target the B1 receptors. So let's think about these B1 receptors and sigma-1 receptors.
And one more small question, Dr. Reshma Shetty. She has asked that a positive antibody test is evidence that the person had SARS-CoV-2 infection. Whether this means person is immune, in or other words, we don't have whether the person is protected from the reinfection, because there are several cases recorded in the reinfections.
Yes. So that is an issue that is going on, being debated. And there have been 2-3 cases, starting the first one from Hong Kong. And then there was a debate about a patient from South Korea. And I don't know about patients. There was one patient in Bangalore that was reported just 1 or 2 weeks ago. Yes, we know that these antibodies, both T cells and B cells are activated. You do get the antibodies, and then IgG vanishes after 45 days or so. And then the T cell activity is memory T cells activity has not been properly tested with in everybody. Cannot be tested. It's very expensive procedure. So what I can say is that right now only few cases. Generally, remember, this is most people are getting cured of the disease in the sense of the infection, and we are able to, the immune system is able to eradicate the virus completely. That means definitely there is orchestration of the proper immune response. But in vast majority, in 20% of the people, there is some hyperactivity of the immune response. So bottom line, I think there is immune response, proper immune response. I feel positive that there is antibodies properly produced and memory B cells and T cells properly produced, but we still don't know why this. But by the way, even when we have the so-called antibody T cells and B cells, memory T cells and B cells, it's okay to have infection. It's not, only it will be subdued. It will not be a full-blown infection. It's normal to have infections. That's nothing wrong. It doesn't say anything negative about the immune response.
With the permission of the chair,
yes, please, sir.
Can I talk to Professor Srinivas? First of all, I would like to congratulate Srinivas for a wonderful presentation. I'm an ENT surgeon, but I could understand a lot of things about COVID virus. It's a wonderful presentation. You made it very simple. I think all of us have been benefited by your talk.
Thank you very much.
Continue this interaction with my faculty and students. I think will be
Absolutely, I love to. Yes.
For particularly we got a medical school with the undergraduate, postgraduate, and PhD programs. One maybe I may be asked to ask this question regarding hydroxychloroquine.
Yes, please.
I understand that the mortality of medical personnel in India compared to the common man has increased. Almost, they said, I could read that around 17 times it's more. And one of the postulation is they are taking prophylactic hydroxychloroquine. Whether it will decrease the immunity of the individual so that they will become susceptible for complications.
Okay. So what I feel that I can say 3 points about hydroxychloroquine. Hydroxychloroquine does have an impact on the immune system because that's the reason why there's a rationale for using it in rheumatoid arthritis. Whether it makes it prophylactic use may be dangerous. I'm not sure about that. And secondly, hydroxychloroquine may not be useful in the context of alkalizing the virion endosomes because we are not having the virion getting into the cells by endocytosis, I think. Supposing that were to be a major mechanism, then hydroxychloroquine could have done the alkalization and could have helped in reducing the viral disassembly. But I don't think that mechanism is not occurring. So the major mechanism for viral entry seems to be ACE2 mediated binding, and then TMPRSS2 mediated cleavage of the S2 site, and then the virus enters, then undergoes disassembly. Is the endosomal route is not very common. So hydroxychloroquine, that's one of the reasons why it is not being effective, because the postulate, one of the main hypotheses is that this hydroxychloroquine alkalizes the endosomes and prevents the viral disassembly, and that is not happening. And that's why we are not seeing the efficacy. And also the concentration and the dosing has to be understood. So are we trying to use it for blocking the viral multiplication? Then what is the dose to be used and where? And is it for blocking hyperinflammation later on? And then of course we need a different dose, I think. So again, these are the things that are not sorted out. And once a scientist said in a recent talk that I heard is that large amounts of data is there that have to be properly distilled, and a report has to be written to understand whether hydroxychloroquine has efficacy or not. And I think the timing of administration of these drugs is very, very critical. So we cannot simply say that use it everywhere and then expect efficacy. It's not going to happen. Thank you very much for that question, because I did not know that you have the challenge of prophylactic use causing significant disease. Thank you.
Shilpa, is there any time for 1 or 2 more questions?
Yes, sir. We can have more questions.
Yes, sir. Sorry, Dr. Srinivas, it is midnight to you.
No, I will be very short. I will be very quick. Don't worry. You can ask as many questions as you want.
No. Because as time passes, there is a lot of jerk in the information flow of knowledge and other things. One Dr. Professor Fatima, she is a NITA University Dean, Nursing Dean. She has asked, is there any proper identification or what? There are several PCR mechanisms. Which one is the best and authenticate?
Oh, I think what I've heard this debate by many times. There is one release from ASM, American Society of Microbiology. I can send her that video discussion. I think there is a massive review that was released recently in one journal. I think this is organized by American Society of Microbiology. And also everybody seems to say that the primers designed by WHO is the best. Okay, so but in any case, one has to stick to, remember everybody says the samples have to be from nasopharyngeal swabs, and you take that and follow their primers and follow the regimen. But the mutations themselves are not affecting the effectivity of the PCR test because the mutations are very minimal.
And there are several common questions or inquiries that one is from Dr. Singh from Bikaner, another one is Dr. Pratap Maurya from CDCRI. They all wanted your presentation slides. I said in the NITA website, NITI website, it is they are going to upload tomorrow, I think.
Yes, please. Yes,
And
yes.
it is
I can send
already
it to
there
you.
recorded in the YouTube. So that
Yes,
with permission of the Vice Chancellor and management and organizing committee, please upload that into that your web page, sir, so that everybody will follow the Professor Srinivas talk.
So fortunately, many of the schematics are drawn by me. So and all the citations.
No problem. That is not a problem. That is
Yes.
not a problem. And many compliments, sir. It's a really inspirational talk. Many compliments are there.
Okay.
So now my question is open to the podium. Anybody is there? Please interact. It's short. Very short. Interact.
I think, sir, we can start with the next speaker.
Okay. Yeah. Okay.
Thank you very much.
Thank you, Professor Srinivas. See you sometimes. And my personal regards to you, personally, because you have a lot of collaboration for publications. One more is coming, scientific reports. Sure. Recently.
Thank you.
Thank you, sir. Thank you, Srinivas, sir. On behalf of
Namaste to all. Namaste to everyone.
Thank you, sir.
Thank you. Bye. Bye.
So we'll move to our next speaker. Over to the moderator.
Yeah. Professor Vimal Karani is in the panel.
Yes, yes.
Yeah. So can I start, Dr. Shilpa?
Yes, sir. You can.
Yeah. So our next speaker in this COVID webinar, international virtual conference, Professor Vimal Karani, yes. He is an associate professor of nutrigenetics and nutrigenomics in the University of Reading, UK, and program director for the food and nutritional science degree programs on the Reading University, UK, and principal investigator and founder of a new startup, Gene Nutrition Interactions Collaborations. He has more than 75 publications in the reputed international journals and 2 book chapters, and supervised the 9 PhD students in such a small young age, and examined more than 7 PhDs, this one. And starting from 2013 onwards, every year he is getting great awards of the Europe. It is really encouraging. One of the most recent award is RSUSU Teaching Excellent Award of the University of Reading, and Celebrating Success Award of same, International Society of Nutrigenomics Young Scientist Award. So many awards is getting. And he is a member of many professional bodies, like member of Nutrition Society UK, member of Nutrition Society India, member of Nutrition Society and Human Genetics, and European Society of Human Genetics. And he's a scientific advisor for the many scientific committees, international scientific committees, like Nutrigenics, Institute of Nutrigenics India, and Velocity Genetics Company India also. He peer-reviewed the many great Nature NPG publication and Cell, Lancet, JAMA, and all other publications he reviewed. Importantly, he reviewed 12 great grant applications since 2014 in Europe. He's editorial board member of Metabolism, BMC Cardiovascular Journal, Oxidative Stress and Cellular Longevity, International Journal of Endocrinology, International Journal of Diabetics and Clinical Research. Yes, several invited speakers, both at the national, international workshops, meetings since 2013 onwards, and more than 33 conferences he attended, and press release more than 20. Now I invite Professor Vimal Karani. Next to you, sir.
Thank you so much for the great introduction, and thanks to the organizing committee for inviting me to deliver this lecture. So let me share the screen with you.
Yeah.
Are you able to see the screen?
Yes, yeah, it is audible and readable.
Okay, great. So first of all, a very good morning to everyone, and also good afternoon to those people who are joining from different parts of the world. And so I'm going to talk to you all about nutrigenetics and cardiometabolic diseases with a focus on COVID-19 and nutrition security. So this talk is going to be totally different from what you have been listening in the last 1 hour, because it's more of an epidemiological perspective. So we know that cardiometabolic diseases are a risk factor for the development of COVID-19. So which means that if you can prevent the development of cardiometabolic diseases, then you can prevent indirectly the development of COVID-19 infection as well. So that is going to be the focus of my talk. So in terms of looking at food security and nutrition security. So what is food security? So the 4 main pillars of the food security are the availability, access, utilization, and stability. Whereas if you look at the nutrition security, it is more to do with the community's access to the essential nutrients. So the food security deals with these 4 pillars, and the main difference or the imbalance that occurs in terms of malnutrition, which is purely because of the food insecure households, when they sacrifice food quality in favor of food quantity in order to avoid the state of hunger. So that is when people are being exposed to malnutrition. And you all know that malnutrition is one of the most important risk factors for the development of cardiometabolic diseases. And there are a lot of studies that have come up in the last few months, which have clearly highlighted the point that people having obesity or diabetes or cardiovascular disease, they are at an increased risk of developing COVID-19 infection. So which means that if you can overcome this malnutrition, then you can overcome the development of COVID-19 infection. So I'm going to explain this talk with regards to the influence of the genetic risk. So with regards to that, I'm going to tell you what is nutrigenetics and what is nutrigenomics. So nutrigenetics deals with the study of the impact of the DNA sequence variations on chronic disease outcomes in response to a particular diet. For example, if you have a very high genetic risk and you are consuming a high protein diet or a high carb diet, what is the risk of developing obesity or cardiovascular disease or diabetes? So in order to answer that question, you do a nutrigenetic study. The nutrigenetic study is where you're trying to understand the interaction between the genetic risk and the dietary factor and how they interact and contribute to the development of a particular disease. Whereas nutrigenomics, as the name implicates, genomics, omics means expression, genomics is gene expression, and nutrigenomics is the impact of the nutrient on gene expression. So gene expression is where the DNA is converted to RNA and RNA is converted to protein. So that conversion of the DNA to protein takes place in every cell in response to the diet that we are consuming. So the diet is inducing the gene expression in your body. So the findings from nutrigenetics and nutrigenomics are essential to implement personalized nutrition. That is where you're developing a diet for an individual according to that individual's genetic makeup. So if I have a low genetic risk, what is the kind of diet I should consume? Or if I have a very high genetic risk, what is the kind of diet I should consume to overcome the genetic risk? So this is what you do as part of personalized nutrition. But to implement such personalized nutrition strategies, you need evidence from nutrigenetics and nutrigenomic studies. So as part of this lecture, I'm going to focus on 2 important nutrients which have been widely and extensively studied in relation to COVID-19. One is vitamin D and the other one is vitamin B12. And we all know that deficiency of vitamin D and vitamin B12 have been linked to cardiometabolic diseases. And also there are studies to show that the deficiency of vitamin D and B12 are linked to COVID-19 as well. So now I'm going to explain the triangular relationship between these micronutrients, cardiometabolic diseases, and how that is being related to COVID-19. So starting with vitamin D. So this is going to be the first part of my lecture. So vitamin D has been shown to be associated with obesity, blood pressure, hypertension, diabetes, cancer, so various chronic disease outcomes. And at the same time, there are studies that have shown that these cardiometabolic diseases are also associated with lower levels of 25-hydroxy vitamin D concentrations. So now the question is, whether is it low vitamin D status that is contributing to the development of cardiometabolic diseases, or is it the cardiometabolic diseases that are contributing or are causing low vitamin D status? So that is what I'm going to explore in the form of an epidemiological perspective. So when you do any kind of epidemiological study, you need to understand the biological pathway by which the exposure is causing the disease. So you need to understand the vitamin D metabolic pathway and how the genes and the environmental factors, they interact and contribute to the development of a particular disease. So if you look at the vitamin D metabolic pathway, there are several genes which are influencing the vitamin D levels. So on exposure to the sunlight, the first step that takes place is the conversion of 7-dehydrocholesterol to pre-vitamin D3, and that takes place with the help of the enzyme 7-dehydrocholesterol reductase, which is encoded by the gene DHCR7. And this gene encodes an enzyme called as a 7-dehydrocholesterol reductase, which converts 7-dehydrocholesterol to pre-vitamin D3. And the next step is the conversion of the pre-vitamin D3 to vitamin D3, and which further gets converted to the major circulating metabolite, which is the 25-hydroxy vitamin D. And this takes place with the help of the enzyme 25-hydroxylase, which is encoded by the gene CYP2R1. So until here, it is the synthesis of 25-hydroxy vitamin D. And the next step is basically the metabolism of vitamin D. That takes place by binding to the vitamin D binding protein, which converts the vitamin D to 125-dihydroxy vitamin D3, which is a calcitriol, with the help of the gene CYP27B1, which encodes the protein alpha-1 hydroxylase. And calcitriol is further metabolized to calcitroic acid with the help of the enzyme 24-hydroxylase encoded by the gene CYP24A1. So if you look at the entire metabolic pathway, there are certain genes which are influencing the synthesis of vitamin D, and there are certain genes which are involved in the metabolism of vitamin D. But these are not the only factors that are determining the vitamin D concentrations. There are also environmental factors, which includes the exposure to the sunlight and the seasonal variation, the geographical location in which the person is living. So all these factors also contribute to the influencing the levels of vitamin D. And of course, the lifestyle, your diet, your physical activity levels, and exposure to the sun, and the place where you're living, and taking vitamin D supplementation. So all these factors do contribute to the variations in vitamin D levels across different people and across different populations and across different ethnic groups. And moreover, socioeconomic status, that is lower and upper socioeconomic status, studies have shown that social context plays a very important role in contributing to the variations in vitamin D concentrations. And finally, last but not least, your personal characteristics, your height, your weight, that is your BMI, your mental health status, your cognition levels, the gender, men, women, your skin color, hair color, all these characteristics do contribute to the levels of your vitamin D. So even though there are several environmental factors, the genetic risk of having low vitamin D level is a very important factor. And in order to overcome that genetic risk, you need to have a moderation of all these environmental lifestyle. That is, dietary modification or the lifestyle modification is required to overcome the genetic risk of having low vitamin D status. So quite recently, I carried out a systematic review with my PhD student where we looked at different vitamin D related genes and in relation to metabolic diseases. And as you can see from the slide, you can see that there are about only 73 articles which are focusing on vitamin D genes in relation to metabolic diseases. It used to be a time when vitamin D was linked only to the bone-related disorders, but now we have moved to a situation where vitamin D genes are also related to cardiometabolic diseases. And in this review, we identified nearly 73 articles which links vitamin D related genes in relation to metabolic diseases such as obesity and diabetes, which means that 73 is a very small number, and definitely more studies are required to establish the link between vitamin D and other cardiometabolic diseases. So this is one side of the story. So what is the other side of the story? How vitamin D is related to your immune system. So we carried out the study almost 10 years ago, trying to establish a relationship between 25-hydroxyvitamin D concentrations and immunoglobulin E concentrations. So in this graph, you can see on the x-axis, you find the vitamin D. On the y-axis, you find the immunoglobulin E levels. And you can see that individuals having normal levels of vitamin D, that is sufficient amounts of vitamin D in their body, they are maintaining a normal levels of immunoglobulin E. But when your vitamin D level goes down, or if it goes higher, then the immunoglobulin E levels show some alteration. That is, they go higher as well. So these fluctuations in the immunoglobulin E levels can be attributed to the differences in the levels of vitamin D. So when vitamin D deficiency occurs, there is an increase in the immunoglobulin E levels. And not only this, we also carried out a genetic study trying to understand whether the genetic deficiency, that is genetic risk for vitamin D deficiency, can that lead to very high levels of immunoglobulin E. So in this graph on the x-axis, you find the vitamin D deficiency and vitamin D sufficiency on the two extreme ends. On the y-axis, you find the immunoglobulin E levels, and the three lines that you see, basically, they are basically low risk, moderate risk, and a very high genetic risk. So you can see that individuals who carry very high genetic risk and being sufficient, they have very low levels of immunoglobulin E. So which means that even if you carry a very high genetic risk, but if you maintain good amounts of vitamin D in your body, then you can still have lower levels of immunoglobulin. So which means that having good amounts of vitamin D concentrations in your system, that can overcome the genetic risk of having high immunoglobulin E levels. But if you look at the other end, people who are deficient for vitamin D, so those individuals, they have very high levels of immunoglobulin E. That is, it's not only the genetic risk, but also low levels of vitamin D that is increasing the immunoglobulin E levels. So which is clearly highlighting the fact that the genetic risk with vitamin D deficiency can increase your risk of having an infection or allergic reaction. So now we have a clear picture of vitamin D, cardiometabolic disease, and COVID-19. So that is, in terms of hypothetical perspective, we do have an understanding of what is happening, but still it is unclear whether vitamin D deficiency is definitely causing COVID-19 or vitamin D deficiency is definitely causing cardiometabolic disease. And the link between cardiometabolic disease and COVID-19 also needs to be established. So if we are able to identify this causal relationship between vitamin D and cardiometabolic disease, and then we will have a better understanding of how we could prevent COVID-19. So that is what I'm going to focus in my talk now. But in order to establish that relationship between vitamin D deficiency and cardiometabolic disease, what we generally do is do an epidemiological study. But doing an epidemiological study is not very easy. For instance, if you want to test for the association between exposure and the disease, for example, vitamin D and cardiometabolic diseases, the main problem is the confounding. So there's always a confounding factor which can affect the interpretation of the association between the exposure and the outcome. And secondly, reverse causation is a very important issue, where it's not only the exposure that is causing the disease, sometimes the disease can also contribute to the exposure. So to overcome these two problems in epidemiological study design, we used a genetic approach, which is called as a Mendelian randomization. So what is Mendelian randomization? So when you test for the association between X and Y, X is the exposure and Y is the outcome, there is always a confounding factor which can affect the interpretation of the association between X and Y. So it is very difficult to establish a causal relationship. So what we do is we use a genetic variant or the genetic variants as instruments which mimics the exposure X. So the causality is inferred from the association between the genetic variant and the outcome of interest. That is, instead of testing for the association between X and Y, we test for the association between Z and Y, so which is a reflection of X and Y. Why do we use genetic associations? Because genetic associations are less prone to confounding and they are free from reverse causation. So, but in order to do a Mendelian randomization approach, you definitely need a large number of samples. So we started off with this large collaborative effort, which is called as a D-cardia collaboration. D-cardia stands for vitamin D, cardiovascular, and diabetes-related traits. To date, we have nearly 35 studies comprising more than 100,000 individuals, and we do have access to various consortia of these studies and the UK Biobank, which comprises 500,000 of the UK population. And in total, we have a sample size of about 2 million individuals. So by making use of these individuals, we have carried out several causal relationships between vitamin D deficiency and various cardiometabolic diseases. So that's what I'm going to share with you all. So the first study that we carried out was basically in relation to obesity, and the genetic markers that we had chosen to study in this particular project was basically these 4 genetic markers. The 2 genetic markers which are involved in the synthesis of vitamin D, and these 2 genetic markers are involved in the metabolism of vitamin D. So basically we studied 4 genetic markers and we tested for the association of these genetic markers with obesity, blood pressure, hypertension, and cancer to identify whether there is any kind of a causal relationship. So starting with the first finding, which is about the obesity and vitamin D deficiency. So there are studies that have shown that high BMI has been associated with low 25-hydroxyvitamin D concentrations. And there are studies which have shown that vitamin D deficiency is associated with obesity. So it's more of a bidirectional question. So we do not know whether is it obesity causing vitamin D deficiency or is it the other way around. So we carried out the study in 42,000 individuals from the D-cardia collaboration, and we identified that for every 10% increase in genetically instrumented BMI, there was a 4.2% decrease in vitamin D concentrations, suggesting that higher BMI was associated with vitamin D deficiency. Or in other words, obesity is causing vitamin D deficiency, but not the other way around. So this is actually the finding that we observed. So obesity, that is high BMI, was causally affecting the low vitamin D status or resulting in the low vitamin D status. And that could be one of the reasons why obese people have very low levels of vitamin D concentrations. And the next study that we carried out was the vitamin D and blood pressure. This is actually a very interesting study because there are several epidemiological studies that have been carried out, and a lot of randomized controlled trials have been carried out as well. While epidemiological studies provided an evidence for the role of vitamin D in blood pressure, whereas the randomized controlled trials failed to show the vitamin D blood pressure lowering effect of vitamin D. So the randomized controlled trials was a complete failure, whereas the epidemiological studies showed an evidence. So we decided to put an end to this controversy and inconsistent findings. So we carried out the study examining the link between vitamin D and blood pressure, and we looked at 155,000 individuals, and we identified that for every 10% increase in genetically instrumented vitamin D levels, there was a significant decrease in blood pressure and hypertension. So these findings clearly highlight the point that taking vitamin D supplementation might be an effective way to overcome cardiovascular disease-related outcomes such as blood pressure and hypertension. At a clinical level, these levels of blood pressure, 0.29 or 0.39, so these are the effect sizes that we observe. So this drop in blood pressure is very, very small from a clinical perspective, but at least this study forms a base for developing the randomized controlled trials, because one of the main limitations in randomized controlled trials is what dose of vitamin D supplementation is required to overcome the risk of developing a blood pressure or hypertension. That dose of supplementation can be decided based on such large-scale studies, and this kind of a study, a Mendelian randomization study, provides a platform for developing such supplementation and the dose. And the third study was done in relation to vitamin D and cancer. So we carried out the study in nearly 70,000 cancer cases and 84,000 controls in relation to 7 different types of cancer, and we used this data from different consortia, and we tested for the association of vitamin D with 7 different types of cancer, and we found an association only with the prostate cancer, where we found that higher 25-hydroxy vitamin D concentrations were protecting individuals from developing prostate cancer. So suggesting a causal link between high vitamin D concentrations and lower risk of prostate cancer. And independent of all these studies, quite recently, I carried out a study which is a phenome-wide association study. So not sure how many people will understand this concept, but just trying to explain that in a very simple way. So there's actually a genetic epidemiological method, which is called as a genome-wide association study. The genome-wide is where you're screening the entire human DNA for different genetic variations which are causing a particular disease. So you're looking at one particular disease. For example, if I want to identify any particular gene for obesity, then I'm screening the entire DNA and trying to identify whether any of the genetic variations are associated with one disease. So that is a genome-wide association approach. Whereas a phenome-wide association approach is where you are screening different diseases and you're trying to identify whether one genetic variant is associated with different disease outcomes, multiple disease outcomes. So we used this Mendelian randomization phenome-wide association approach to examine the causal relationship between BMI and multiple disease outcomes. So we looked at a genetic instrument for BMI and we tested for the association with multiple disease outcomes. That is, we looked at nearly 925 disease outcomes, where we had a sample size of nearly 1.7 million individuals with data from UK Biobank and different other consortia. And when you run such large-scale association analysis, you get a plot, something like this, and this plot is called the Manhattan plot. This is just to show you that we know that obesity has been shown to be associated with different disease outcomes, but we do not have a clear evidence whether obesity is causally associated with different disease outcomes. So that is a very big question. So this study, which was published, where we published last year in Lancet, where we clearly identified that obesity was causally associated with 33 disease outcomes. So out of 925, we filtered it to 33 disease outcomes, which includes not only diabetes or cardiovascular disease, but also it included several infectious diseases and also leading to endocrine diseases and various other skin diseases. So obesity is a very chronic problem and also an inflammatory disease leading to multiple disease outcomes. So this is actually a clear identification of the fact that obesity is being linked to COVID-19. And this study clearly identifies that obesity increases the risk of infections as well as part of a causal relationship. Because showing an association is different from showing a causal relationship, because causality is the one that is required from the point of pharmacogenomics or pharmacogenetics in terms of designing drugs and how to treat any particular disease. So these are the kind of studies which are required in terms of implementing or developing or discovering new drugs for new diseases. So all these studies that I've shown you so far, they've all been carried out in European population, that is focusing mainly on the Caucasian population. So what is happening in lower middle income countries with regards to the vitamin D studies? So I carried out the first nutrigenetic study in relation to vitamin D in the Asian Indian population. So just to highlight the study. So in this graph on the x-axis, you find the carbohydrate intake, low, medium, and high consumption of carbohydrate. On the y-axis, you have the 25-hydroxy vitamin D concentrations. So you have the white bar and the black bar, which is the low genetic risk and high genetic risk respectively. So if you focus on the low carbohydrate consumption group, you will see that individuals consuming low amounts of carbohydrate and having a low genetic risk, they have significantly higher levels of vitamin D. So in order to have a high levels of vitamin D in your body, it is not sufficient you just have a low genetic risk, but also you need to consume a healthy diet, which is something more like a low carb diet. So this study clearly identifies the fact that even if you are low genetic risk, even if your genetic risk is not high, still you will have to consume a healthy diet to make use of that low genetic risk so that you can get protected from the development of vitamin D deficiency. So not everyone who have a low genetic risk can be protected from vitamin D deficiency. It doesn't work that way. Even if you have a low genetic risk, you still need to consume a healthy diet to overcome the genetic risk of vitamin D levels. So this study clearly identifies a very important solution for people who keeps asking, I have a very low genetic risk, so can I eat whatever I want? Definitely not. If you want to make use of your low genetic risk, then definitely you should consume a healthy diet. Only then that low genetic risk will protect you from developing the disease. So the next study was done in the Indonesian population. So this is just to show you some of the pictures of how the South Indian population is consuming. They do consume significant amounts of carbohydrate and a lot of sugary foods, indicating the fact that they need to reduce the carbohydrate consumption. That is, their intake of carbohydrate is like more than 500 grams of carbohydrate per day. So definitely reducing it to less than 300 grams per day can overcome the genetic risk of 25-hydroxy vitamin D concentration at a lower level. So now moving on to the next study, which is the study in the Indonesian population. So this is actually called as the vitamin D pregnant mother study, VDPM cohort study. So the whole idea of the study was to look at the pregnant mothers right from first trimester, second trimester, and third trimester. And we also measure the vitamin D concentrations at the first trimester and the third trimester. And we also looked at the anthropometric measurements of the newborn babies. Our question is whether vitamin D deficiency during pregnancy period, whether that has got any impact on the newborn anthropometric measurements. So there were two phases of the study. In the first phase, we wanted to know whether pregnant mothers had anyone reported of low vitamin D status. So we carried out the study. So in this graph on the x-axis, you find the low and the high genetic risk. On the y-axis, you have the vitamin D concentrations. And you can see that individuals or the pregnant mothers who have a very high genetic risk, they had significantly lower levels of 25-hydroxy vitamin D concentrations, especially during the third trimester. So which is highlighting the fact that if you have a high genetic risk, you're likely to have lower levels of 25-hydroxy vitamin D concentrations. So now the next question is, what is going to be the outcome of these pregnant mothers who are vitamin D deficient during the third trimester? So in the second phase of the study, we identified a very interesting finding, that is the pregnant mothers in the third trimester who had a very high genetic risk and those who had low levels of vitamin D, they gave birth to the babies who had a very small head circumference. So in this graph, you can see that on the x-axis, that is a normal head circumference and a smaller head circumference. On the y-axis, you find the 25-hydroxy vitamin D concentrations. The white bar is a low genetic risk and the black bar is a high genetic risk. So if you focus on these individuals, you can see that mothers, the pregnant mothers who are vitamin D deficient during the third trimester and who had high genetic risk, they gave birth to babies who had very small head circumference. And you know very well that if the babies had very low or small head circumference, that becomes a risk factor for the development of cardiovascular diseases at a later stage. So this study clearly highlights the fact that irrespective of the age and irrespective of the gender and irrespective of like whether you are young or old or in the middle age, it's very much important that we all need to maintain sufficient amounts of vitamin D concentration. And especially during the development, that's what we say, the first 1000 days. that is very crucial for the development of various physiological aspects in your body, and that requires sufficient amount of all these micronutrients. So in terms of looking at the take-home message from all these studies, it is very clear that the vitamin D plays a very important role. And in fact, some of my studies clearly highlighted a causal relationship between low vitamin D status and a high risk of cardiometabolic diseases, including cancer and various other disease outcomes. So in terms of looking at food security or nutrition security, looking at the four pillars, availability, access, utilization, and stability, it is very much important we need to understand where the problem is. Even though we have the availability is not an issue or access is not an issue, and stability of the availability of the foods and access to the foods, that is also fine. But in terms of utilization, if individuals are not living in an area where the sunlight exposure is very, very less, then the best way to overcome this problem of vitamin D would be to take vitamin D supplementation. So taking vitamin D supplementation and having sufficient amounts of vitamin D levels in your body, that can help you to overcome the risk of developing cardiometabolic diseases, and indirectly you can overcome the risk of developing COVID-19 or becoming a victim of COVID-19 infection as well. So making vitamin D supplementation as a mandate is not present as a kind of a policy at the moment, but that's something that needs to be implemented in different ethnic groups, in different populations, in order to make sure that everybody is vitamin D sufficient and not deficient. So this is the aspect in relation to the vitamin D and cardiometabolic diseases in relation to the COVID-19. But looking at the next level of the nutrition security, which is the vitamin B12. So we know that the main source of vitamin B12 is all the seafoods and various other meat-related products. And not only that, also the dairy products, eggs, and other seafoods. So deficiency or not getting access to all these food products can lead to the deficiency of vitamin B12, resulting in cardiometabolic diseases. But you have to understand that living in a country such as India, or especially if you take the South Indian population, most of them are vegetarian, and that is one of the reasons why we have a very high prevalence of vitamin B12 deficiency. So looking at the relationship between B12 and cardiometabolic diseases, B12 deficiency has been shown to be associated with obesity, diabetes, and cardiovascular disease. But at the same time, there are studies which have shown that these cardiometabolic diseases can also contribute to lower levels of vitamin B12. So it's a question whether is it the B12 that is causing these cardiometabolic diseases, or is it the high risk of cardiometabolic disease that is lowering your B12 levels. So in order to explore this relationship, of course, you have to use an epidemiological study design. But as I said before, in order to examine the association between exposure and the disease, the two main problems are the confounding and the reverse causation. So how do we overcome this problem again? So what I did was I used a genetic approach to understand the relationship between the diet and the disease. And the diet, what I'm talking about, is a B12 nutrient, and the disease is basically the cardiometabolic disease outcomes. So looking at the relationship, so I tested for the cardiometabolic genes with cardiometabolic outcomes. I tested that association. And independently, I also looked at the B12 related genes which are lowering the B12 levels in relation to the B12 deficiency. And next, I looked at whether cardiometabolic genes were associated with B12 status, and likewise B12 genes with cardiometabolic disease outcomes. The reason why I did that is basically this will give a better understanding of the relationship between cardiometabolic diseases and the B12 deficiency. So I used a genetic approach to understand the relationship between cardiometabolic disease outcomes and the B12 deficiency. And then I also tested whether adopting a healthy lifestyle, that is consuming a healthy diet and doing more levels of physical activity, can that overcome the genetic risk of B12 deficiency and cardiometabolic diseases. So I'm going to give you some examples of the study that I've carried out in relation to the B12 and cardiometabolic diseases. So I started this genuine collaboration. Genuine stands for the gene-nutrient interaction 6 years ago. The main objective is to implement the nutrigenetic studies in contrasting ethnic groups. And these are the countries in which I've implemented the nutrigenetic studies. And these are some of my publications. And these are the different funding organizations which have supported my work. So the main objective of this genuine collaboration is whether any particular nutrient is interacting with your genetic risk and contributing to the development of the disease. So the first example that I'm going to give you is basically the relationship between B12 status and the metabolic disease in this Asian Indian population. So we collected about 2,000 individuals from the city of Chennai, and we tested whether anyone with a genetic risk, do they have lower levels of vitamin D concentrations. So in this graph on the x-axis, you find the genetic risk for the metabolic diseases. On the y-axis, you find the vitamin B12. The red bar is high metabolic genetic risk, and the blue bar is a low genetic risk. And as you can see, individuals carrying very high genetic risk, they have significantly lower levels of B12 concentrations. So having known that if you are having a high genetic risk, it means that you're going to have low levels of B12 levels. So then the best way to overcome that would be knowing your genetic risk and taking vitamin D supplementation or improving your vitamin D rich food consumption. And the second study was done in the Sri Lankan population, which is another South Asian population. So here we tested whether the genetic risk for B12 deficiency was associated with central obesity. We looked at the waist circumference, the waist-hip ratio, on all these parameters relating to central obesity, but we did not find any association with central obesity. But when we tested for the interaction with the dietary intake, that is, we found an interaction with the protein intake. So individuals who are consuming higher amounts of protein-rich foods, they alone showed a higher risk for central obesity, which is what we call as apple-shaped obesity. So which clearly shows that the risk for central obesity is not independent, but the risk is being conferred to the central obesity under a dietary influence. So your diet plays a very important role and how it interacts with the genetic risk and contributing to these cardiometabolic diseases such as central obesity. And the third study was done in the Indonesian population. Here we focused on a very interesting community, which is the Minangkabau community. Minangkabau is the world's largest matrilineal community, and we looked at Indonesian women living in this population. This is a very interesting finding. On the x-axis, you find the tertiles of dietary fiber intake. On the y-axis, you find the glycated hemoglobin, which is a risk factor for type 2 diabetes and an indicator for type 2 diabetes. And the blue bars are the low genetic risk and the red bar are the high genetic risk. And if you look at the individuals consuming very low amounts of fiber-rich foods, you will understand that people consuming very low levels of fiber-rich foods and carrying a high genetic risk, they have significantly higher levels of HbA1c. So what it means is that if you're consuming low fiber food, which means that it's an unhealthy food, you're consuming your very low amounts of fiber-rich foods and you're increasing your carbs or increasing your fat intake. So which is obviously an imbalance in your nutrient intake. So consuming low levels or unhealthy diet is increasing your genetic risk for higher HbA1c level. So increased levels of HbA1c becomes a factor for type 2 diabetes. So your diet is actually your determining factor. If you are able to consume good amounts or healthy amounts of fiber-rich foods, then you can overcome the genetic risk. So if you look at the high consuming group, there's no impact on the HbA1c level. But whereas here, consuming unhealthy diet is aggravating your genetic risk for higher levels of HbA1c, which can eventually lead to the development of type 2 diabetes. So in summary, if you look at all these studies, the diet plays a very important role. For example, in this Indonesian population, they do consume significant amounts of fried foods. They use a lot of oil. And in fact, like most of the studies have shown that they do consume significant amounts of coconut oil in their cooking and meat-related products. But even though they're consuming significant amounts of meat-related products, the way they cook and how much of the B12 is being consumed by these individuals is a very big question. So in summary, if you look at these three traditional diets, the first one is the Indian diet, and the second one is the Sri Lankan diet, and third is the Indonesian diet. So these are the traditional diets, and the traditional diets are considered to be quite healthy, and they are being consumed like for several decades ago. But now what's happening is that due to the urbanization and westernization of the dietary patterns, that is increasing the genetic risk and leading to various cardiometabolic diseases. So the main conclusion of the story is that the genetic risk of B12 deficiency can lead to metabolic diseases only under a dietary influence, which has been clearly seen in South Asians and Southeast Asians. So now going back to that take-home message from the B12 studies, of course, there is a link between B12 and cardiometabolic diseases, but that is clearly seen only through the dietary influence. So the stability is very much important. The availability and access to the B12 related foods, that is very much essential. If you're not able to get such foods, it's important that you need to take the vitamin B12 supplementation if you're not able to get access to any of the B12 rich foods, because that alone can prevent the development of cardiometabolic diseases. And if you're reducing the risk of cardiometabolic disease, which means that indirectly you're reducing the risk of becoming a victim of COVID-19. So if you're interested in knowing more about the genuine collaboration, feel free to go into this publication, which I published early this year in the Proceedings of the Nutrition Society. So besides South Asian and Southeast Asian countries, I'm also looking at different parts of the world, different lower middle income countries, not only in Asia, but also in the western part of Asia and South Africa and North Africa, and also the West African population and the Middle Eastern population, and also in the South American population, independent of what I'm doing in all the developed countries. So in line with it, I would like to thank all the DCARDIA collaborators, and thanks to all the genuine collaborators as well. And finally, don't forget to maintain the social distancing and also to follow a healthy living pattern. And most importantly, if time permits, do get your genes tested and blood tested to know whether you are vitamin D deficient or B12 deficient. And if you know that you have a deficiency, please do take these supplementation. That is very important, because one of the main limitations in lower middle income countries is that we assume that we are healthy unless we get to know about all the symptoms relating to vitamin B12 and vitamin D deficiencies. So don't leave it to the last minute. So get your blood tested, and also if possible, get your genes tested, so that if you know your genetic risk, you can overcome the development or the risk of developing cardiometabolic diseases. So on this note, I would like to thank you all once again for listening to my talk. I'm happy to take any questions. Thank you.
Yeah, can I start, Dr. Shilpa?
Yes, sir. You can
Yeah.
start with your questions.
Yeah, so very nice presentation, Dr. Vimal Karani. Really inspiring and motivational one. It's really good eye-opening for the COVID management through the lifestyle and food management of the personalized food management. So I have some questions in our chat box.
Yes.
One is Dr. Fatima De Silva. In case if the individual has genetic alteration associated with the vitamin D deficiency, will supplementation of vitamin D helps in the individual or not?
Okay, yeah, thank you so much for an interesting question. So if you take the vitamin D genetic variation, so vitamin D metabolic pathway is very complex. If you look at the systematic review that we did, we looked at about like 4 or 5 genetic markers. And these 4 or 5 genetic markers are contributing only to about, say, 20 to 25% of the variations in the vitamin D concentration. So which means that nearly 75% of the remaining variations need to be identified. So it's very difficult to say that if you have a genetic variation, whether the vitamin D supplementation will work or not. So unless you look at the entire genetic variation that are available from the human DNA in relation to the vitamin D outcome, only then you can have a clear picture of whether vitamin D supplementation will work or not. So every individual has got like different levels of risk, low risk, a moderate risk, and a very high genetic risk. And it also depends on which category you belong to. If you know that if you have a very high genetic risk, then you can overcome that genetic risk by dietary supplementation or, dietary modification or by vitamin D supplementation. Yes.
One more question from Nelly Mayallu. Professor Vimal.
Sorry, I'm not able to hear you.
Yeah. One more question from the Nelly Mayallu.
Yes.
How we changes this genetic risk for the reduced their risk of obesity according to your research, sir?
Sorry, I didn't get the question. How do you change the genetic risk?
Genetic risk for the reduced their risk of obesity. But that means genetics, obesity, how it is interconnected to reduce that obesity directly from the food management. I think that is the theme of the questions, core of the questions.
Yeah, so that is where actually this concept of nutrigenetics comes into picture. The whole point is, once you know that you have a very high genetic risk for obesity or any chronic disease outcome, then how do we actually overcome that genetic risk by adopting a kind of healthy lifestyle? So when I say healthy lifestyle, I'm not talking about the universal dietary recommendations. I'm talking more about like what could be the best diet for your DNA. Because my DNA is different from your DNA, and your DNA is different from that of others' DNA. So everybody is unique in our own way. And not only at an individual level, but also at the ethnicity level, at a population level. And that is one of the reasons why, like, you can easily identify, okay, this person is a South Asian, this person is from South Africa, this person is a Caucasian. How do you identify that? Because it is their DNA which is contributing to their physical representation, which is what we call as a phenotype. So the kind of the dietary modification that you adopt should be done in relation to your ethnic group or at the individual level. So that can overcome the genetic risk of obesity.
So can I take some more?
Yes,
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 sir,
sure.
you have 50 more minutes, sir. You can take many more questions. I think there are many questions coming up.
Oh yeah, one more question from the earlier speaker, Dr. Srinivas. He says that generally thought that vitamin D is involved in the gene expression vis-a-vis immune system, non-specific. So non-specific treatment, will it help or not?
I didn't get the question. So what does that mean? Where is the question coming?
Yeah, I think Dr. Srinivas can directly ask.
Yeah, so I was wondering, in America, we don't prescribe vitamin D or zinc supplements or vitamin C supplements to patients, COVID patients, when they get admitted to the hospital or when their positivity is known. So what is the significance of urgent, what is the significance of administration of vitamin D just after diagnosis of COVID patients? Yes.
Oh yeah, so that's exactly the crux of my talk. So I didn't go into like the vitamin D having a direct link with COVID-19 because there are a lot of randomized control trials which have shown a positive impact of vitamin D supplementation on suppressing the COVID-19 symptoms. But at the same time, there are some studies which have failed to show a positive or significant effect. So that could be due to various reasons. And some people require high doses of vitamin D supplementation, and some people require moderate doses of supplementation. And you can clearly see that there is a difference in the way people are responding to the vitamin D supplementation. And these responses, these differences in the responses can be clearly attributed to their genetic makeup and their genetic risk. So I don't believe in a kind of a universal therapy. I believe more in looking at the individualized therapy, because I believe in a way by which you can treat an individual in a personalized way. And that is exactly how these several drugs are working. There are certain drugs which will work for some people, some diabetic patient, and there are certain drugs which will doesn't work for some other diabetic patient. And likewise, inflammatory bowel disease, there are certain drugs which works very well for these patients and not for other people, and which is purely the underlying hypothesis behind pharmacogenomic therapy as well, because the DNA is different and you require any diet or drug which is unique for your DNA.
Thank you very much.
Yeah, thank you.
So one more questions from Dr. Trupti Kainar. How long we can give vitamin D supplement to patients? Once it stops, again, there is a deficiency observed in the patients. Kindly suggest, sir.
So your patients, by means you're talking about COVID-19?
I think it is general one. If Trupti is there, she can ask directly. I think she's not there. Anyway,
Yes.
I think it is muted. I think it is general questions, not COVID directly.
Okay, so generally, like when you take this vitamin D supplementation, it takes like 6 months to absorb like good significant amounts of vitamin D in your body. So it's good to do your blood test, and if you find that you have sufficient amounts of vitamin D, then you can take, you can stop your supplementation course, and it depends on which part of the world you're living in. For example, I know some of my colleagues who live in Finland, they do take vitamin D supplementation almost every other day because they don't get sunlight exposure at all. So it depends on which part of the world. And you don't have to be a patient to take vitamin D supplementation. It could still be a part of your daily routine supplementation that you could take on a daily basis.
Thank you. One more question from Pooja Patil. Do we get vitamin B12 by plant source?
Yes, of course, like because that is how the vegetarian people who are consuming vegetarian diets are surviving, I suppose. And at the same time, like of course, B12 from the plant source is quite less compared to the non-vegetarian foods like meats and seafoods. And that is exactly the reason why we are doing a project like where we are trying to compare the South Asians living in India and South Asians living in the UK and comparing that with that of the Caucasians living in the UK, and we are trying to understand why there is a significant prevalence, because the prevalence of vitamin B12 deficiency is about like more than 20% in the Indian population, whereas it's less than 10% in the UK population. Of course, UK people, they do consume significant amounts of meat-related products.
So I think Shilpa, we have time, no?
Yes, sir. Can I ask one question, sir?
Yes.
Yeah.
Does age matter with this supplementation or the level of vitamin D?
The age.
Yeah. How age plays a role in this?
Yeah, generally, like I mean, vitamin D deficiency and B12 deficiencies are generally considered under the chronic diseases and more often adult onset diseases, because for a genetic variation to show an impact on a chronic disease outcomes, it requires sufficient time or chronic exposure to the differences in the environmental conditions. So definitely vitamin B12 deficiency and B12 deficiency, they are more related to the adult onset diseases such as obesity and diabetes. So just because like you're deficient today, you're not going to develop the disease tomorrow. So it takes a long time. And of course, if you find that children are having vitamin D deficiency and B12 deficiency, that could be an outcome of various other complications. So it needs to be consulted with a doctor to check whether the levels have got any other implications.
Yeah, thank you, sir. I think Sucheta ma'am has one question.
Yes.
Very nice presentation, sir.
Thank you very much.
Hello, very informative. So what about probiotic supplementation for vitamin B12?
Sorry, probiotic?
Probiotic, yeah.
Yes, there are. Yes, so when talking about the probiotics, that is more related to your, because I didn't go into that aspect about your microbiome. That is another interesting area of research, because the probiotic supplements and fiber-rich foods are very much essential for improving the production of all the short-chain fatty acids, and which can have a positive impact on your gut microbiome and also different health outcomes. And studies have shown that taking probiotic supplements and fiber-rich foods can increase your B12 levels in your body as well.
Thank you, sir. One more on vitamin D. When we expose to sunlight maximum, the intake is reduces.
Sorry?
When we expose to maximum to sunlight, is the vitamin D intake reduces?
Vitamin D intake means, you mean vitamin D absorption reduces?
Vitamin D requirement will be reduced if you expose to more sunlight, because cholesterol, 7-dehydrocholesterol can also become active form.
See, one question that goes on every time, like that's being questioned to me is, I'm getting exposed to the sunlight on a regular basis, but still, why am I having very low levels of vitamin D? So that is always a very big question. And one of the main reasons for that is your genetic susceptibility. So in India, vitamin D deficiency is highly prevalent. Why? We do have sufficient amount of sunlight exposure, but why are we having a very high prevalence of vitamin D? And also in terms of looking at the dietary intake, vitamin D from diet is very, very less. So definitely it can be attributed not only to various other physiological conditions, but also to your genetic susceptibility and your genetic risk. So when your genetic risk is very high, even if you're exposed to the sunlight, you can have very low levels of vitamin D concentration. So that is where I was talking about the utilization plays a very important role from the food security point of view.
Thank you very much, sir.
Yeah.
A great information.
One more. Can I take, madam?
Yes, 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 sir.
Yeah. One more question from Professor Indrani Karunasagar.
Yeah.
All the vitamin D and B data on levels is from the Western studies. What is your experience with the Indian, Sri Lankan, and Indonesian community?
Well, I think that's what I presented. Like now
Yes,
we have
yeah, it is
evidence
there.
from the Indian, South Asian, Southeast Asian population. But the main challenge is basically if you had looked at all the data from the Western part of the world, I had like huge, massive, large sample sizes because in the developed countries, developed part of the world, they have already collected lots of data, and we have like UK Biobank and different consortia have been started, and we do get loads and loads of data set, and I have like a sample size of about 2 million individuals. So you can do any kind of a relationship or a study, association study to examine like how vitamin D can have an impact on cardiometabolic diseases. But if you come down to the lower middle income countries or any developing countries, the main challenge is to have a well-characterized cohort. So if you take any study population or any data set that is available, they might have data on cardiometabolic diseases, but they might not have data on vitamin D. Whereas some data sets will have information on vitamin D, but not on cardiometabolic diseases. So that is actually one of the main limitations I had. So what I had to do was I need to start a new study in each and every population. I had to collect the data right from the scratch to get all the information and do the study. So that was one of the biggest challenge. And that is one thing like that needs to be improved in every population, because in terms of epidemiological studies, the lower middle income countries, they are lacking in various resources, and in terms of funding, we do not have sufficient funding, and that limits us from having large scale population-based studies.
Thank you. One more question from Vishnuvardhini Datta. She is, I think, from VIT University, Vellore. She says that there are a lot of vitamin supplements are available in the OTC, and people just go and purchase during the COVID pandemic. What do you suggest about this a mad rush towards the vitamins supplements during this pandemic period, sir?
I think like that would be, it would be better if a clinician answer that question.
Yes.
But from my point of view, at least you need to take like 1,000 units of vitamin D per day in order to meet your vitamin D requirements. Because like from dietary intake, as I said before, from dietary intake, you're not going to get that much of vitamin D. And of course, you need sunlight exposure. And during the lockdown, that was actually a very big issue in terms of going out and getting exposed to sunlight. So that is one of the reasons why people would have gone to different medical stores to get all the vitamin D and other micronutrient supplements.
There are some questions on my WhatsApp box from the University of JK, Jammu Kashmir. He says that environmental condition and vitamin D, does it have any direct correlation with the immunity? That means in Kashmir, you have most of the time snow and...
sorry, can you increase your volume? Sorry.
Yeah. There is one question from the JK, Jammu and Kashmir. He says that there is a lot of environmental variation around the year. So consuming this vitamin D in summer, winter, and autumn and spring, does it have any effect on the chronic ill patients or on the COVID?
Yeah, because seasonal variation always plays a very important role in influencing your vitamin D levels. So as mentioned in my slide, where I showed the genetic influence and also the environmental influence, and where I mentioned that one of the most important environmental factor that is influencing your vitamin D levels is the seasonal variation. the seasons plays a very important role. In fact, like we had published a paper showing that especially during winter in the UK or in western part of the world, the vitamin D level goes down, and during that time, people are more prone to develop different infections. And that is one of the reasons why in the UK, especially during the winter season, people develop cold and flu and various other viral infection, and that could be clearly related to the study that I mentioned about the vitamin D concentration and immunoglobulin E concentration. So that plays a very important role. Of course, it does have an important role to play.
Sorry, you are not tired with the volume of the questions, because it is interesting talk. Is it okay? Yeah. One more question. One more question from the Banaras Hindu University, Varanasi, where Shraddha Saroj is asking: is there any harmful effect of higher levels of vitamin D than the normal in case someone continuously takes supplements?
Sorry, what is the
She's asking, I think she's asking, is there any harmful effect of higher level of vitamin D than the normal in case someone continuously takes supplements? Continuously takes.
Yeah. So as mentioned in the study that we published almost 10 years ago in the journal Allergy, where we were comparing the vitamin D status with the immunoglobulin E levels. So as long as you maintain sufficient amounts of vitamin D in your body, or not getting into deficiency or excess of vitamin D in your body, so the excess levels of vitamin D can increase your immunoglobulin E levels in your body, so which is indirectly increasing your infection levels as well. So what you need is, taking too much of anything in excess can become an harmful impact on your body.
True. Sorry, my subject is nano formulations and regenerative subjects. I have one personal questions. What is your thoughts on the nano prebiotics for the probiotics?
Sorry, what is the thought about?
The nano prebiotics for the probiotic enhancement in the gut microbiome.
So definitely, like the probiotics and the prebiotics are essential for maintaining the healthy gut lumen and the microorganisms. And these are the ones which are actually stimulating the release of all the short chain fatty acids, and also maintaining the immune systems in our gut lumen. So that is definitely highly recommended for all the people who have a very low immunity, and improving. And especially during the lockdown, you would have seen, like I was coming across a website where they were trying to sell different kinds of probiotic supplements, and chemically engineered all these probiotic supplements which are trying to improve all the gut microbiome. But I'm not sure like whether they are being tested in terms of kind of a randomized control trial, or any of the testing has been done in humans, because especially during the lockdown, these have all been released. So not sure how many of these will work. But I'm not sure whether this Dr. Praveen Jacob, is he there on in the session? If he's there, he'll give you a better understanding of how the natural way of improving your immune system. He talks very well about this probiotic supplements and prebiotic, and which have got a very beneficial effect.
Dr. Praveen is here. Dr. Praveen Jacob is here.
I don't think so, sir.
Yeah. Okay. No problem.
Okay,
No problem.
Okay, I'll inform you. Okay.
Yeah. One Dr. Reshma A. Shetty, I think she is from NITE. She has asked. Yeah, she has asked that we have n number of epigenetic modifications. Do you think only healthy diet can overcome this?
Yeah, so there are two important aspects. One is actually the genetic, and the other one is epigenetic. So when you talk about genetic, it's more to do with your genetic risk. Whereas when you talk about epigenetic, which is considered as a non-genetic mechanism, it has got nothing to do with your DNA sequence. You're looking at the gene activity in epigenetics. So epigenetic modifications can be reversed with your dietary modification, whereas the genetic variations cannot be modified. If you're born with high genetic risk, until the last day of your life, you have to live with that genetic risk. Whereas the epigenetic modification, they occur during your lifetime, and it all depends on the environmental influence, and your epigenetic modifications can be reversed definitely by dietary modifications.
So now over to MC. Is there any questions or anybody
Yeah,
from
I
the
think
podium?
we have utilized the time that was
Correct.
there. So we'll move to our next.
So one minute, I will
Yeah.
thank you. Thank you, Professor Vimal, and looking forward for the fruitful and continuous collaboration with the Nitte University and my university, Miyazaki. Thank you.
Yeah, thank you so much.
Thank you, sir. On behalf of Nitte University, we had a very great talk. Thank you, Vimal. I'll call you back.
Yeah, sure. Okay, thank you so much.
Yeah, I once again thank our speakers, Dr. S.P. Srinivas and Dr. Vimal Karani, for a thought-stimulating session, and also our moderator for conducting the whole session smoothly. Next, we have lined up a panel discussion on the topic live feedback from recovered patients about hospitalization, treatment, and post-COVID life. The moderator for the session is Dr. Vadish Bhatt, Professor, Department of ENT, Care Medical Academy, Nitte deemed to be university. Now I request Dr. Vadish Bhatt to take over the session and introduce our panelists for the day.
Good morning, respected faculty. Good morning, respected faculty, delegates, and my dear friends. At the outset, let me congratulate the Sakura Science Forum for organizing a very apt one-day conference on COVID-19. I appreciate the enthusiasm of the team in organizing this virtual conference in spite of the prevailing tough situation. COVID-19 pandemic has caused unprecedented impact on human life, and the life of most of the world population had come to a short pause. However, medical profession is an exception, as there was no such pause seen in the life of medical professionals, but in fact they had to work hard and differently from the earlier days. There is a change in the way we live, we examine, and we care for our patients. However, the attitude too has been changing over a period as we started to live with the coronavirus. Also, apart from fighting with the virus and the disease, healthcare workers had to fight against the ignorance of some of the people sometimes. During this process, many of our healthcare professionals across the globe got infected, and unfortunately, few had to suffer the maximum. By now, most of us have gained some knowledge and education about COVID-19 with the help of research papers, media, and the social media. However, this panel discussion is special, for there is nothing like listening to the words from the horse's mouth. Today, we have with us three of our faculty from Case Medical Academy representing the pool which they belong to, who are actively involved in the diagnosis and treatment of COVID-19. Dr. Giridhar BH is the professor and head of the Department of Pulmonary Medicine at Case Medical Academy. He is also the member of the COVID Task Force and also the COVID Expert Committee of Justice Case Charitable Hospital. He is one of the faculties who are closely monitoring the COVID situation in our institution. I welcome Dr. Giridhar for this discussion. Dr. Amit Kilgi is the assistant professor in the Department of Microbiology who had a special training in COVID-19 diagnosis at the National Institute of Mental Health and Neuroscience, Bangalore, NIMHANS. He is also the nodal officer for COVID-19 diagnostic lab and also the member of the COVID task force. He is also the infection control officer of our hospital. I welcome Dr. Amit for this panel discussion.
Thank you, sir. Good morning, everyone.
The third panelist is Dr. Ganesh, who is also a microbiologist of Case Medical Academy and Justice Case Charitable Hospital. He is involved in the virology laboratory at the Kshema, taking the technical responsibility of performing the RT-PCR. Welcome, Dr. Ganesh, for the discussion.
Thank you, sir.
Keeping in mind the theme of the conference, let me start from yesterday. When SARS-CoV-2 virus entered India, we fortunately had little knowledge about the behavior of the COVID-19 by observing the events in other countries in the East and the West. In Mangalore, Kesegde Hospital is among the first to get geared up to face the situation. Dr. Giridhar, as a member of the COVID task force, can you share with us briefly what was the response of our institute and how good were we initially to face the situation? What was the immediate focus of the COVID task force and what were the measures taken to segregate COVID positive and COVID suspect patients from non-COVID patients? Dr. Giridhar, please.
Good morning to everybody. Thank you for this opportunity for the forum. So it was a sense of fear of unknown in the beginning of the March because that time we started hearing so many cases being reported in India, and the Kasaragod, which is just 60 kilometers away from the Mangalore, has become a kind of hub of coronavirus, which had 100 cases at that time, which was a big number. So we are all geared up, and especially our institute has been extremely proactive in supporting the task force. And they had a one clear agenda that none of our faculty should suffer because of this. So whatever the precautions, whatever the logistical supplies required, you go ahead. And also management was very clear that because this is a pandemic, even though there was a talk of government hospital being set up exclusively for the COVID care, but management is of the very clear opinion that one day we have to step in. So when we have to get into that mode, so better to prepare as early as possible. So we got all kind of support. In fact, they were extremely courteous to extend many of the facilities to us which were not existing, which I'll enumerate in due course. So this was the time during the lockdown is being implemented in the countrywide in a sequential manner. So there was no lab in the Mangalore. We had to send samples to Hassan, which is more than 200, close to 200 kilometers away, and the turnaround time was minimum of 3 days because they were also burdened with the samples from most of the places. So we started from there, and in the 23rd March, I guess, we had the first case of SARS-CoV-2, which was in a 10-month-old infant, and which was the first infant to be reported from India at that time. So it was the first infant. So suddenly there was a sensitization throughout the hospital that yeah, we are stepped into the territory of corona, and there is a the sensitization was instant. So that helped us, and as a task force, which was also formed during the May 18th or 19th. So immediate task was to lay out what are the protocols, how to face the scenario, how to segregate the COVID and non-COVID patients. And again, there was a treatment aspects also. There was a big buzz around the hydroxychloroquine as a preventer of the infection. Even also some of the, it was advised in a prophylaxis for healthcare workers. There were ICMR guidelines was suggestive of that. So we wanted to make the availability of those things. And then the PPE, the personal protection kits, equipments, which were, because we never thought that it has to be geared up in such a situation. And when even the institute was ready to procure any amount of PPE, which was not there in the market, the Prime Minister also has repeatedly stressed that we were having very few small production of the PPE, but today we are the second largest production in the world. That's how even the whole apart from the healthcare, the many other sectors also responded. And in the initially we had to find it very difficult to get the PPE, but some of our faculty members who are very active went to the extent of requesting some of the factories to open and place advance, and we could get the PPE in as and when required. So I never had any deficiency of PPE because the many different areas were been set up. The severe acute respiratory illness ward, stable suspect ward, confirmed stable ward, again stable, and ICU, again suspect ICU, as well as the confirmed ICU. So all these things we had to look after. And then there is the fever clinic, which is a pivotal in the management of these patients, because segregation of the patient into COVID and non-COVID was important. We were running a small fever clinic, which was a screening area. But as in the demand increased, we felt that this is not enough, because there will be some ambiguous patients, which we will not be able to diagnose at the first shot whether it should go to COVID area or the non-COVID area. So we expanded that to a 10-bed facility where patient can be waited for an hour or two till the investigation report comes in, X-rays is done, even ultrasound abdomen, every investigation, every procedure could be done except the aerosol generating procedures because the precautions is required. So we made a fever clinic almost like a small emergency room, and the decisions are taken based on the investigation reports and the clinical assessment. And that helped us to segregate the patients quite well to the COVID and non-COVID area, and which also helped us to prevent the unnecessary exposure of the healthcare staff, because it is not possible to be work in PPE in all the areas. It is very difficult. We have donned and doffed PPE multiple times. Still, I feel people who wear PPE for a longer durations, especially 6-hour shift, and it's very, very difficult. So it's not possible to put PPI everywhere. So we had to strengthen our fever clinic. We did that, and we could able to secure so many COVID patients into the COVID area because of our very active fever clinic. And OP triaging area we set up. Because of the OP triaging area, there was a double check. Once the OPD started, there was a triage area. Any suspect patients, any doubtful patients would directed to the fever clinic, and fever clinic would work up, and then it's segregated to the this area. So here I would like to stress also, some of our doctors are infected, but we are the least infected in the city compared to other hospitals. And I owe this to the fever clinic, and everybody pitched in, they took the responsibility. And because of that, our infection rate of the healthcare workers is less than the others. Then we had to increase some of the infrastructure also, especially the COVID ICU. We needed some extra ventilator that was provided. We needed a high frequency nasal oxygen devices that was provided. And then we also needed an RT-PCR lab, because RT-PCR lab was set up in the Mangalore, and still there was a delay, and there was a approval whether this, even though we felt this patient can be COVID, and we need to have an approval from the infectious disease surveillance officer before sending the RT-PCR. So to do away with all these things, we had to set up our own RT-PCR lab, and our management and the microbiology team and many others, many other in central research lab maybe, and even the NUXAR, and many people have involved and set up a RT-PCR lab. And after setting the RT-PCR lab, the turnaround time became very less, and we could diagnose even in a patient is in suspect area, within 1 day report will come, so we know whether it's a positive or negative. And that helped to helped greatly in the management of these patients, because if there's a 3-day turnaround, means we have lost crucial 2 days in starting maybe some of the antivirals and all those things. So the RT-PCR lab was an important thing which made our management aspect also much easier. So I stop there.
Yeah, so that is quite impressive. The task force action, the quick action from the task force was commendable, and they have shown that there is always a way if there is a will and willingness. Now you mentioned about the RT-PCR lab. Of course, that is a very important landmark event happened during the COVID period. So Dr. Amit, the testing for COVID-19 was a challenging task in the early days, because we had to send that and transport the collected samples to the government laboratories and wait for the report, which used to take few days. Sooner, Shema had set a virology lab, as mentioned by Dr. Giridhar. As you are actively involved, and of course you are among the pillars of the virology lab, can you briefly share your experience in the challenges in setting and functioning of the lab?
Yes, sir. Thank you for this opportunity to speak on the panel. We earlier, as sir said, used to send samples, send it to Hassan, which delayed a lot of reports and management. Then we start, then along with the management, we showed willingness to start the RT-PCR lab. We sent our letter to the ICMR and the government. Then they said, yes, we can start the PCR lab. Next step was to get trained for it. So we, team of 4 went to the Nimhans to get ourselves trained to basically see how to set up a PCR molecular lab for testing COVID, and also to see how the processing works. Then after coming back from the training, next crucial challenge was to set up the molecular lab from the scratch. We did not have a molecular biology lab in the diagnostic area. So with a lot of help from the management and various research institutes like our NUCSER and ERL, we were able to set up the molecular lab from scratch within a very short period of time. So they had a great management and our senior support who put all their efforts so that we could start the lab as early as possible. Next important hurdle was to get the NAB, accreditation board approval, which is the board which mainly sees the quality of it. So getting an NABL accreditation is quite a lengthy procedure, but with the tireless and day and night efforts of our team, we were able to get the NABL accreditation for quality, and all the paperwork done in astonishingly 10 to 15 days. And we went for accreditation and finally got the accreditation in the end of the May, got the ICMR approval to start the lab. And since then we have been successfully functioning and running the lab, testing samples round the clock, and are able to deliver the reports within 24 hours. And we are not only testing our samples, we are also catering to the government samples. We are getting the reports to them as early as possible. Since there is a huge load of samples from the government, we are still able to deliver the reports and cater to the needs of the patients.
That is a great achievement indeed, to have a virology laboratory set up in a very short time. Of course, I can say a record time, because our laboratory was otherwise not NABL approved earlier. So Dr. Amit, as the days rolled, there were a few new investigation modalities introduced. Can you share information about other modalities of diagnosis like rapid antigen testing and antibody testing apart from RT-PCR?
Yes, there are various testing modalities, and the gold standard being the RT-PCR. Similar to this molecular method, we have another two, that is CBNAT and TrueNAT. They are nothing molecular methods only which detects the RNA, but those two are cartridge-based systems. So, and advantage is that it can deliver the results faster. But advantage is only a few samples can be tested at a time. So when with a huge load of samples, it is difficult to use those setups. So PCR is the best way to deal with a huge sample load, and it's the gold standard. The down the lane, we got other testing modalities, that is the rapid antigen test, also called as the rapid point of care test. The point of care test is the magic word here. That is one which simplified some of the problems associated with the delayed reporting in various setups. So this is basically detecting the antigen in a patient who might have got infected. It is a very simple and easy procedure which gives results within half an hour to 1 hour after sample collection. The test has a moderate sensitivity but high specificity. That means that if the test comes positive, then the patient has the infection. There is no doubt in that. Next, if it turns negative, then especially in cases where the patient is symptomatic, then we have to do a repeat PCR and confirm the diagnosis. So that is why we say it is high specificity and moderately sensitive. So rapid antigen test, the government and ICMR started including in its testing strategies, and recently we got on September 4th the 6th version of the testing strategy, where rapid antigen test is given as the first choice of test for mainly surveillance in the containment areas. And also it is also option in case of healthcare setup and in areas of non-containment areas where asymptomatic individuals are to be tested. It is as a choice, and in symptomatic individuals, the RT-PCR is the choice. The advantage, as I said, we get results very fast. The disadvantage is that the sensitivity is low. Again, we need to do a rapid, RT-PCR in case of negative symptomatic individuals. The testing, the ICMR says that repeat testing is not advised, which is actually followed. So the ICMR says that repeat testing is not required once a person is positive by any of these testing modalities. Only if a negative person again develops symptoms, a repeat testing is advised. Otherwise, if clinically the person is fine, then there is no requirement for repeat testing. Then next we had the rapid antibody test. So that actually started off with a boom in the market. ICMR only recommends CLIA and ELISA method of antibody testing. It is basically testing the IgG antibodies in a person who has been infected and the body has mounted an immune response to that. These IgG antibodies start to appear around 2 weeks after the onset of illness. So detecting these antibodies just show that the person has been exposed and his body is able to produce or mount an immune response in an adequate manner. So that is why the ICMR strictly recommended it only for surveillance purposes and not for diagnostic purposes. So surveillance in the community and also testing for the healthcare workers, whether their antibody levels are fine to again prioritize their workings. Also now since the plasma therapy started, it has been used to detect the amount of antibodies, which is one more of the important criteria for screening the donors for plasma therapy donation. So this is antibody testing, which is being used currently. Apart from this, the RT-PCR stands the gold standard.
Yes, thank you, Dr. Amit. The message taken, the gold standard is still the RT-PCR, and antibody is for surveillance, and antigen is for the rapid testing because it has a high specificity. Thank you very much, Dr. Amit, for elaborating on the testing modalities. Now, Dr. Ganesh, you are among the team taking the technical responsibility of performing RT-PCR since the day the virology lab was installed in our hospitals. So we understand that RT-PCR is not a simple procedure, but requires a lot of patience apart from having a good knowledge. The laboratory was new, so was the disease. Can you share with us what was the challenges faced during the RT-PCR testing? Dr. Ganesh.
Sir, unmute yourself.
Yes, sir. So first of all, I thank the organizers for inviting me for this panel discussion. So as sir has told, what challenge we faced, the greatest challenge we faced was providing the reports to the anxious patients. Now, as Dr. Amit has told, the RT-PCR, this is a very specific and sensitive test to detect the coronavirus RNA, the sample, and the test is also a laborious and time-consuming process. Now, this also requires skilled technical staff to process the sample and also to perform the test, and even require experienced staff for interpretation of the results. Now, this procedure involves several steps. Now, there is a sample processing where we will be segregating the samples into individual samples, that is of symptomatic cases, and then go for the pooled samples where each contains 5 samples each. And at a time, we can go for 94 samples, and it takes around 2 hours for doing the sample processing. And then followed by this is RNA extraction, where again, it takes place around 2 hours time to do this, and followed by the RT-PCR, the procedure, and then analysis. So again, this takes around 2 hours. Now, when the individual test report is generated, and it is mainly, it takes around 6 to 7 hours after receiving the sample. Now, again, in this, if there is a pooled sample which is a positive, Again, another 6 hours is required for generating the reports of the individual samples in the pool. So, yes,
thank you, Dr. Ganesh. We do understand that RT-PCR is not a child's play, and performing an RT-PCR is not as easy as just speaking about it. Thank you for elaborating the testing methods. Dr. Giridhar, you are a member of the, am I audible? Sorry. Yeah. So you are a member of a COVID expert committee. So we would like to know from you what were the various presentations of COVID patients in our setup and how the treatment was planned and executed.
Thank you, Dr. Vadish. So we saw all kind of patients, I must say. Initially, when the fear of the COVID was very high in the society, we started seeing a lot of cases from the June 2nd week or so. That time there were a lot of asymptomatic mild cases, so very few serious cases, and there were moderate cases. We could see some of the pneumonia cases and all those things, and with the timely intervention they did quite well. But after late, what's happening is the current, as on today, current India's recovery rate is 77%, and the vast course of people who recovered and went without much intervention, they have spread the word that it is a simple disease, it will be cured easily, you don't have to go to the hospital also. So that's what we get, a lot of calls from the distant places with a mild weather, come to hospital or not come to hospital. So because of this, now what we are seeing is a more of a severe cases we are seeing. By the time they present to us, they are already having a severe disease. Now we are hardly seeing any mild or moderate cases. So the kind of presentations, that is a change in the trend, maybe because of the experience the public has got with this disease. But when we come to the presentation, we have seen as it's described in the literature, we have seen a frank acute myocardial infarction presenting to the casualty. Initially, we thought that it's a clear-cut MI case, but later turned out to be a COVID. So there are few cases initially, we felt that it's a typical cardiac case, but it was secondary to COVID. So later we became a little more strict in screening these patients. And then we saw scores of pneumonia patients presenting with a COVID as expectedly. But we also have seen some of the patients presenting with a cerebrovascular accidents. No other comorbidity, no other illness, but they just presented with a cerebrovascular accidents. Even we have seen a case with the toxic epidermal necrolysis. We don't know whether it's because of COVID primary or it is due to some secondary drugs. So we have seen varied kind of presentations as described in the literature, from a mild asymptomatic large pool of patients to currently severe cases. And now there is a stream of patients who are coming with a acute renal failure and a diabetic ketoacidosis, the worsening of the diabetic ketoacidosis or so.
Sorry, I didn't unmute. So thank you, Dr. Giridhar. As you had aptly mentioned, initially you had to treat some of the ignorance of the patients along with treating the actual patients. So Dr. Amit, we have seen a number of healthcare professionals getting infected with the virus. Generally, a doctor or any healthcare professional takes maximum precaution while treating a COVID positive or a COVID suspect patients. What is your view on how a healthcare personnel getting infected?
Thank you, sir. So we have seen that around the globe, many of our healthcare personnel are getting infected during their fight against the COVID. Now, we healthcare workers are in the forefront fighting this disease, and we have the close proximity to the patient and the infectious person. And there is a high viral load. That is one which is the greatest risk for the healthcare personnel. There are many factors which contribute to increased infections in the healthcare workers. One of them is this high proximity. The other one will be inadequate use of personal protective equipment, and very few instances of, as in early days, less availability of PPE. Even though inadequate use of PPE is a risk factor, but infection has been also seen care workers who had a good compliance in the and a proper use of personal protective equipment. So along with, we cannot blame just on the PPE. There are many other factors also which contribute to the increase healthcare worker getting infected, like many infection control practices which needs to be followed. We are fortunate that we have a very strong system here and good availability of PPEs and masks. So, as Sir said earlier, we have a very less healthcare worker infection in our institute. So mainly the best way to reduce these healthcare worker infection is to strictly follow the infection control practices and see that the infection does not spread to the healthcare worker or vice versa.
That indeed is opening the minds of us. So thank you, Dr. Amin. So to echo what I had mentioned, I would like to mention an interesting paper published in American Journal of Infection Control in August 2020. So that was not a huge study, but in 50% of the cases, they could not trace the contacts. In 25%, the infection was from the hospital, either from the patients or the colleagues. And in 25%, it was from the either family members or the community. And even in the hospitals, what they had mentioned is, interestingly, so among those who getting infected from the hospital, the chance of getting infected was least in intensive care unit, whereas it was higher in the other areas. So the message probably, as you have mentioned, to all the healthcare personnel is, please take appropriate precaution even beyond your work area. So thank you for enlightening us on that area. So now let me come to the point. Dr. Ganesh, so you are among the healthcare personnel getting infected by the virus in the initial days, and hence having the experience of both sides of the table. Can you share your experience as what was your symptom and how did you get yourself tested? And most importantly, what was your reaction to a positive test?
Yes, sir. So Justice K. Sekade Charitable Hospital is providing COVID testing facility in the microbiome department since May for the public. Now, COVID task force under the leadership of the Dean, the MS, and Dr. Vikram Shetty and his team are managing the COVID crisis very well in our hospital. In July, one of our colleagues presented the signs of COVID-19. So as a precautionary measure, the COVID task force took a very good decision to screen all the healthcare workers of the microbiome department who were involved in the COVID testing. So I was also screened for the coronavirus. Now, at the time of swabbing, I was asymptomatic. Now, coming to my reaction, what was my reaction to this? My reaction was a surprised one because as a microbiologist, I was very much confident that I would be tested negative for the virus. Since we see to it that we follow hand hygiene after every step of procedure performed in the laboratory. Now, added to this, the COVID-19 safety also included, that is SMS, social distancing, wearing the mask, and use of the sanitizer. Now, but when the result came, it was positive, which was an unexpected one. Now, the many thoughts crossed my mind. How, where, from whom, when, why me? So that was the reaction I get. Now, how did I contract the infection? That is, how did I get? Now, this is a very billion-dollar question. Now, as I was one of the healthcare worker in the virology lab, maybe that is a possible source, but or I might have contracted from the community itself. For example, in a visit to a barber, a vegetable vendor, or a visit to the grocery shop and so on. Now, why I'm telling this is, even though we had followed the SMS, we still might get infected if other fellow members in the society do not follow them. The message is everyone should follow SMS regularly, whether you are infected or not. That is, do not be a source of infection to the community that might include individuals with particular infection. Sir.
So there was some internet issue. So that is very true, Dr. Ganesh. Even if you are a perfect driver, you can't help if someone comes and hits your vehicle. Now you opted for a hospital stay, even though you had an option of home isolation as per the protocol when you got infected. So how was the isolation procedures in the hospital? And most importantly, how did you spend the time in the hospital? Because you are a busy man, always busy with the virology lab or in the teaching. So can you share your experience as a patient in the hospital?
Yes, sir. So I was given option of home isolation or isolation at the hospital. Now, as I was residing in an apartment, I felt that I should not be a source of infection to my family and the inmates of the apartment. Now, so I decided to get myself isolated in the hospital. Now, again, here the COVID task force of our hospital made arrangements for my comfortable isolation stay. I was advised a 10-day isolation in hospital as I had no symptoms at all. Then on 11th day, a swab was taken, and then they asked me for 4-day isolation, home isolation. In the second swab, it was negative. I was told to report back to duty as per the guidelines for the recovered healthcare workers. Now, the next question was, how did I spend my period in the isolation hospital? Now, the facilities at the hospital was very good. Doctors and the nursing staff regularly attended to my needs and were inquiring about my health from time to time. Now, they advised me to take good rest and drink lots of hot water. Now, if any complication symptoms, they told me to immediately inform the nurse. Now, as I had academic commitments, I was preparing PowerPoints for online classes and also conducted Viva for students. So this was how I spent the period, so that I was not focusing on this COVID infection. But there was some small fear. First 2 days, I had the fear. I was just feeling myself for fever or any cough, everything. Now, in my break time, I was just pondering what may be whether I had symptoms before. Now, when I retrospectively thought, I had mild symptoms. That is mild sore throat, cold, retroorbital pain was there. And then in the hospital, I came to know that I had I experienced loss of smell and also loss of taste. So this was how I spent my period of isolation in the hospital, sir.
Thank you, Dr. Ganesh. So you have spent the time in a most fruitful way, even though you were hospitalized.
Yes.
Now, Dr. Amit, you too were sailing in the same boat as Dr. Ganesh. So can you share your experience on the other side of the table?
Yes, sir. Unlike Ganesh sir was asymptomatic, I was mildly symptomatic with low-grade fever and mild sore throat, which normally happens in this season. So I was also not actually expecting it to be a COVID. But I voluntarily got tested myself to see that I make sure that the people around me do not get infected, because working in a virology lab requires round-the-clock attendance here. So I got tested and unfortunately it came out to be positive. So that was a shocker to me, but since I was mildly symptomatic, I accepted it and got my family members also tested, since they are in close contact, and all 4 of my family members turned out to be positive. So I, with my family members, opted for home isolation. So the life changed. It was a complete different experience during these 17 days of isolation at home. It was a daily challenge at home. All of us were mildly symptomatic, though symptoms were very mild. We had I have a 2-year-old son and a 65-year-old father-in-law who are this category. So it was every day a small symptom or a change in the symptom alarmed us a lot. But in these situations, we had a great support from our Kshema family. They helped us a lot to get through the situation. And as prescribed by our doctors, we had all the monitoring of SpO2s and temperature and any change in the symptoms. So we were asked to, that is what we are expected to do in home isolation. So we all did that and constantly reported to it. And our government also kept track of our health. I hope it has been done in all the areas. Then one thing I realized during the isolation period was that the society reacts differently to the COVID family. I had a completely unexpected experience of people reacting to a positive COVID family, even though it was not a, some part of it was not good experience. Though people are educated, still they have many wrong thoughts about the disease in the community. All played a crucial factor, and I would like to point out that even though COVID affects the lungs and has the cytokine storm affecting the immune system and various organs, it does have definitely a big psychological component. The isolation itself is a big psychological stress. Apart from that, as I said, the society experience is one big. You are locked down, your whole building is locked down, people around you are scared to see at you, and so all these things do play a important role in the patient's recovery. The most important part when a person is in isolation is the support from the family, from each other, and the society is very important. I had a great support within family. My wife is who is also a microbiologist. We all had determined that we will stay at home and see that we will get through this situation without any problems. Then we had support from our landlord, who was, we are really fortunate to have him. He supported during our isolation, getting us whatever we required, and definitely our institute support. So all these support will help you to get through the situation easily when you are especially at home isolation and you have risk groups involved. Otherwise, I want to say that COVID disease is not such a big dreadful monster. For an individual who is healthy, who doesn't have any comorbidities, for that person, he doesn't need to be scared. Only thing everyone has to be is to be precautious, see that they try their maximum, follow the SMS and not get infected and spread the infection to others. Otherwise, they don't have to care so much of the disease. Infection, it will come, it will, if we go for the various seroprevalence studies, we'll see that quite high seroprevalence is there because everyone gets infected. Like we had Ganesh sir here, he was asymptomatic when he was tested. So that infection would have come and would have gone also, and if it comes, rather it will be mild. Only in few cases where comorbidities are associated, only there is a worry of severity. Otherwise, the support, society support, family support, psychological support. That is why our psychologist and our psychiatry department also kept on working for the betterment, and a supportive service was also provided by our institute for COVID patients who were in isolation. So all this together makes a great impact on the lives of COVID patient. Before got the COVID infection, the life was completely different. I was trying to diagnose the disease. Then next was to experience the disease, experience various symptoms, many symptoms, mild symptoms like anosmia, loss of taste, smell, sense, gastrointestinal symptoms like diarrhea were there. Great amount of myalgia. Even after that, we were when we do stops. So all that will be there. So I would like to say that disease per se is not so serious. You can you can get through easily, but required support from the society and the people around you.
Thank you, Dr. Amit, for your reassuring words. Now, both of you have raised a few valid points. Some are contrasting and some are like echoing each other. So the message is the support from family and society is very much essential for complete physical and mental recovery. Also, the protecting the family is utmost important to avoid the unnecessary stress if they also get infected, because then you will be in real soup. So that is why I remember somebody mentioning aptly the healthcare personnel are warriors who take their family to the war front. So thank you for sharing your views. Now back to Dr. Giridhar. Now the latest in Kshema, of course, now a bit older relatively, the plasma therapy is emerging topic of discussion. Of course, it is there in few places for past few months. Recently, the Kshema is ready with the plasma therapy laboratory and the plasma convalescent plasma therapy. So what is your take on plasma therapy? Because there are so much, a lot of speculations and discussions going on about plasma therapy. Now, what is your view regarding the candidacy for donor as well as the recipient?
Yeah, thank you, Dr. Vadish. Yeah, like many other things, like how the RT-PCR lab was one of the first to be set up at Kshema, the plasma therapy unit is the first one to be set up by any institute in the coastal Karnataka. We take that privilege. All kudos to our blood bank team and many helping hands working with that. So we have already transfused more than 100 units of plasma to our patients. Earlier, we used to get it from a private lab in Bangalore. Now we have our own plasma bank. We are getting it from there. So whenever it is used in the moderate and severe patients, it does makes a difference. So our study has to come out of our findings, but this is an anecdotal experience. We seem to feel that it makes some difference, definitely on a positive side. So regarding the candidacy, moderate and severe disease is earlier in the course, it's better. But whenever not many options are left and plasma therapy as such, other than a mild transfusion related reactions, it is not harmful. It will anyway, it will have some proteins and so any antibody titers are checked before giving the plasma transfusion. So it is definitely not harmful. So we can always use it whenever we have not left with much options, because we are no specific treatment yet for the COVID. So it's been useful in our experience in many patients. And regarding the donorship, any person who has had this disease and who has been recovered from it, and after 28 days of first positive report, he can donate the plasma, provided he has a optimal antibody titers. And plasma donation can be repeated after 2 to 4 weeks interval. And this has been done by some of our colleagues who had COVID infection. They came back and again donated a couple of times. So advantage of this plasma therapy, because we follow the apheresis technology, because of that, what happens is only the required plasma is collected. The remaining blood components like RBCs, WBCs, all these, and the other voluminous things are being put back to the patient circulation. So only plasma is being extracted. And for the same reason, as we use apheresis kit, it has been little expensive. The procedure itself costs around 8,000, 9,000 rupees to the, including all the other things. So our hospital is doing a no loss, no profit basis for the benefit of the patients. So that's about the plasma therapy.
Thank you, Dr. Giridhar, for briefing about the plasma therapy and clearing some of the clouds around plasma therapy. Now let me hear something from the horse's mouth again, because two of our panelists, Dr. Ganesh and Dr. Amit, both have donated plasma. So Dr. Ganesh, as a convalescent plasma donor, how was your experience? Can you share with us?
Thank you, sir. Plasma donation was a very beautiful experience for me. It was a great opportunity for me to give something to the society. I decided to donate plasma, and would donate again if required. I am thankful to the plasma therapy and the blood bank team who took care of me during the plasma donation procedure. On the day of the plasma donation procedure, they advised me to have a good breakfast and a lot of fluids. The procedure went on smoothly, which was of 1 hour duration. Now, in my opinion, there is nothing to fear of plasma donation procedure. Just a little niggling feeling would be experienced at the site of needle insertion. Now, this plasma donation, by doing this, I feel thrilled and proud that my plasma is going to save a soul. Now, in this hour of great need, it is our duty to reach out to save lives. I proudly admit that I have done my bit to the society for a great cause. I appeal to eligible donors to come forward in large numbers to donate. This is my humble request. Thank you, sir.
That is great to hear from you, Dr. Ganesh. Dr. Amit, being a plasma donor, what was your experience? Would you like to add something or share your views?
Thank you, sir. Plasma donation experience Ganesh sir shared. Mine is also similar to that. In fact, plasma donation when started in our institute initially was one among the few first ones to donate the plasma. And when was approached to, and mine is a AB positive blood group, so I mean it's a bit not so easily available. So when I was exposed, and I actually had been waiting for someone to ask me for donation of blood or plasma since I used to donate regularly during my UG days, but I was told since you're AB positive, don't keep donating unnecessarily. When asked for, you donate. So I was actually waiting for someone to ask me to donate. So, and then I got this golden opportunity and I was approached by the blood bank personnel. Yes, we require an AB positive plasma. Are you ready to donate? So I was like, I'm more than happy to donate. At least make the use of getting the corona infection and save someone else. So I agreed to it happily and went ahead with the plasma donation. And it is nothing to be scared of. It's a simple procedure, just a needle prick is that you feel. And then the machine does, and your plasma is separated and other components are given back to you, so you will not feel that weakness which you expect. So it is a great experience, satisfactory experience. I can say that once I have donated, and it is always there in my mind that yes, I was able to help one more person in need, and that would have saved one life. So that feeling is you cannot match it with anything else. And I would urge, like sir said, all the convalescent person who got infected and are recovering to come forward voluntarily, register yourself to donate the plasma. And no, don't get scared seeing the machine. It is a harmless machine, just makes few beeps. That's it. Otherwise, the blood bank person actually takes care of you very well. They keep you engaged, and you will not even feel that you are there for an hour there. So please free your minds with whatever doubts you have regarding plasma donation. It is be scary. It is going to affect me, or I might get any other complications related to this. There are very hardly any, or very one in million complications for plasma apheresis. So my urge is for everyone, please come forward, register yourself for plasma donation, and help one more person in need. Thank you.
Thank you, donors, for being proud of your donation. We too are proud of you. Hope there will be many more inspired by you and many lives are saved. Now, something about tomorrow. In spite of the fear of COVID-19 being continued, we do have a ray of hope. As Dr. Giridhar rightly mentioned earlier, India has a very good recovery rate, crossing 77%. Dr. Giridhar, you have already mentioned it. Does it mean that those who have recovered from the disease are they safe? Do you anticipate any delayed complications or sequelae in those who have already recovered? If so, what is your strategy for this?
Yeah, thank you, Dr. Vadish. As on today, we have 3.5 million recoveries in India, that is 35 lakhs, and less than a million active cases. So total cases is around 47 million, 4.7 million, but the active cases are around only less than a million. But we have already a recovered population of 3.5 million. So which is a very nice thing, and with a recovery rate of 77.88%. But there are post-COVID complications is being reported, and we have to be ready for that. And we are seeing a lot of them actually, in our experience too. In post-COVID, the recovery, especially in asymptomatic and mild cases, it's usually a very good recovery. But however, in a moderate and severe cases, who are in some kind of support system like prolonged oxygenation, especially ICU stay, ventilatory support, and who are recovered from the ventilator, we are seeing some crippling complications, like they require prolonged oxygenation, there is a neuromuscular weakness, something like critical illness, myoneuropathy, and also there's a lung fibrosis issue is there, and some of the recovered patients, they are coming with a cardiac rhythm disturbances, and we have seen some of the sudden deteriorations during the post-COVID period. And also, this is one of the most hypercoagulable disease. We have seen patients on anticoagulation in a supranormal doses for months together, having a clots while you are doing some procedures like central insertion and all. This is in our experience. So this is a hypercoagulable disease. There is a lot of Western reports saying that patient who are recovered presented with some thrombotic complications. And also, since this critical cases, they stay in a ICU for a prolonged period, they need a mental support also. There's something like ICU psychosis, and recovery phases are not very good. And there's a loss of family members. Sometimes it's very unfortunate. And unfortunately, in this disease, what happens is, even though when somebody is critically ill, unfortunately, multiple people in the family are also admitted in the hospital at the same time. So there are a lot of socio-demographic issues in this disease. So we are expecting a lot of complications to come, especially lung fibrosis, prolonged oxygenation, and myoneural weaknesses, and cardiac related issues. So thinking all of these ahead, and again, the Justice K. Segde Hospital takes a privilege of setting up the first comprehensive post-COVID care clinic in this part of the country, in the coastal Karnataka. We are the first one to set up. It's already up and running. We are extending our services to patients who have suffered from the COVID-19 disease and have a perpetuating health issues. This comprises of an elaborate panel of doctors, including specialists from the various departments, and the particularly internist, and from the pulmonary medicine department, from the cardiologist and the psychiatrist. Also, it involves chest physical therapy unit, even yoga and breathing exercises, and psychiatrist also. So we want to extend, or we are very clear about, we are focused about extending the services beyond the recovery, which is needed, and we are focused on that.
Thank you, Dr. Giridhar. So comprehensive post-COVID care, it's a great concept indeed. Yes, apart from the internist and the pulmonologist, psychiatrist and psychologist do have a greater role in this setup, as already experienced by Dr. Amit and Dr. Ganesh. So your words mean that you are very well prepared for tomorrow's situation too. But let us not stop the discussion only at tomorrow. I want to hear from you something for the coming days in future, and of course, for the coming generations too. Dr. Giridhar, many of the delegates in this conference are the scientists and researchers. You are also among the faculty of our university who are actively involved in research. We have faced one greatest challenge in our lifetime in the form of COVID-19, and the need is to convert this challenge into some of the opportunity in the form of research and contribute to science and the healthcare for the benefit of the mankind. Can you share with us how our institution and the university is involved in research related to COVID-19?
Thank you, Dr. Vadish. our university, especially our Vice Chancellor, Dr. Satish Kumar Bandari, has led from the front in preparing us for the research related to the COVID-19. He has been very instrumental, and he insisted to focus us on the research works. So with his inspiration and the suggestions from the dean and all, our institute is extremely focused on the research related to the COVID-19. Some of our faculty members are working on the genomics related to COVID-19. Some of are working on the proteomics, and some of the researchers are focused on the clinical outcome and ICU outcomes, and our faculty members are involved in the ICMR related research. Already our one of the faculty is being chosen for that, and there are many intramural funding options are explored for the researchers from the various faculty levels. So I don't go to the divulge of what exactly we are doing, because it is not a mandate for me. However, many of our faculty are involved and focused on this research, and there is a research care cell has been focused so that there is no duplication of the research, and also there is a for the smooth transition between the clinicians and the researchers, and there is so many things have been set up so that even when we are taking consent, multiple consents have been avoided through a ethical committee approval. So university and the hospital and the researchers are all focused on this thing, because we are not only looking at the treatment, but as you said, we want to look at the research outcomes, so that the not only today, even in generations to come, there should be a contribution from the NITA University for the betterment of the care of this SARS-CoV-2, and we are very much into it, and I am proud of it, the kind of support extended by the university in that aspect too.
That is great to hear about the research activities being conducted in the NITA deemed to be university. As Professor Dr. Madhyastha mentioned in his inaugural address, there is a lot of opportunities for collaborative research in COVID-19. And even the other speakers, Dr. Srinivas SP and even Dr. Vimal, too have stressed upon these collaborative research activities. I feel we need to consider this message seriously and execute the same. Dear friends, though there is an uncertainty in everyone's mind as when the things are going to be normal and when we can go back to our earlier normal situation, let us hope and make up our mind to go for an improved version of our life rather than the earlier pre-COVID world. The fear of COVID-19 should not stop us from setting new goals for a better tomorrow. Our panelists have shared their experience both as diagnosing and treating physicians and as COVID patients recovered. I thank all the panelists for joining us and sharing their views and the experiences. I thank the organizing committee of the conference, Professor Fatima and Professor Sujata Kumari, and Professor Harish Madhyastha, and the members of the Sakura Science Forum for giving us an opportunity to be a part of the conference and share our experience and the views. I thank Dr. Srinivas SP and Dr. Vimal Karani for being present throughout the discussion, even though it is odd time for them. So we appreciate your support and encouraging us, sir. I hope we could do some justice to the responsibility given to us. Thank you all.
Thank you.
Thank you, Dr. Vadish Bhatt and our panelists. I once again thank our panelists, Dr. Ganesh, Dr. Amit Kelly, and Dr. Giridhar BH for their valuable time and a very provocative and informative discussion session was it. Now we have come to the end of this one-day virtual conference on COVID-19 pandemic, yesterday, today, and tomorrow. But before now, may I request Dr. Sucheta Kumari N, Member Secretary, Sakura Science Forum, to deliver the words of gratitude.
Good afternoon to everyone. I'm feeling very proud that we have initiated this virtual conference by the grace of God, and our Vice Chancellor has made us to do this virtual conference. And but I called Dr. Madhyastha sir to help us. And in between, there are a lot of hurdles. We supposed to complete this conference on August, but getting speakers was and in time from and the relevant speakers was very difficult. And meantime, I was also told by VC to arrange this panelist for the discussion of those who are infected cases in this discussion. So that was also, we were really struggled. Finally, I got Dr. Vadish Bhatt, Girish sir, and Amit sir, and Ganesh. So all these with the speakers, our Srinivas sir and Vimal, we could do this virtual conference with a great success. So this is at the end, I have to show my gratitude to all of you, and our mainly I have to show my gratitude. It is my privilege to propose vote of thanks in this occasion, that is on behalf of Nitte Deemed to be University and the University of Miyazaki and Nitte Sakura Science Forum. I must thank our university officials. That is very important for us because it is a university program. Mainly to our Honorable Chancellor, Sri Yanvi Hegde, and Pro-Chancellor, Dr. M. Shantram Shetty is the Pro-Chancellor for Hospital Management, Mr. Vishal Hegde, Pro-Chancellor for Admin, and our Vice Chancellor. Already he was presided the virtual conference, presiding officer, plus he was every day inquiring how was the progress is going on. So university, our Dr. Harish Madhyastha sir was every minute, he was waiting for the progress, and he was the one who introduced us our main speaker, that is SP Srinivasan sir. And then he said, Sucheta, you only find one more speaker. Then I was fortunate to attend Dr. Vimal Karani's talk in one of the virtual conference. I was really felt he is our right person to select. So I contacted him and we gave them separate date, and everyone date was fixed, time was fixed. Then I was searching for this panelist discussion, which was very, very important. And VC was every day calling me, who is, I was searching, I was going through everyone. And finally, God's favor, I met Dr. Vadish Bhatt, Ganesh, and Giridhar sir, and Amit sir. Those are willing to spare their valued time, all that made us to success this conference. So I am very grateful to all the university officials. And in addition, our registrar, Dr. Alka Kulkarni, our Director, Dr. Indrani, Dr. Deputy Director, Dr. Praveen, and all the heads of the institutions, our Kshema Dean, Dr. Prakash, Vice Deans, and everyone supported directly or indirectly for the success of this conference. Then I have to give few more gratitude to our eminent speakers and our own Indian speakers. Those are settled USA, Dr. S.P. Really, I'm very grateful in the coming years for the support for the research and other support for the research students also. And Dr. Vimal Karani is a young scientist Fortunately, I met him through the virtual conference, and really we are very grateful to you, and on behalf of the Nitte University, we are thanking you, sir, and we need your support for the future research collaboration. So then our own person, Dr. Haris Madhyastha sir, since so many years is helping us to support for the research as well as for the development of the Sakura program. We are very grateful to you, sir, your presence, your support. There is no words to explain. So throughout the conference, even early morning today, 6 o'clock, sir, madam, sir was calling me whether everything is right. So we are planning, and I'm doing this type of conference so many times. So I think we are all the troubleshoots we know advance, we prepared for the all types of troubleshoots, and for all the troubleshoot we are away from the troubleshoot by the panelists, even though they four are all the doctors with very busy schedule, and Vadi sir had today some program, but still he made his time, and with his team, you know, the end all the programs end with the entertainment like that. They given wholeheartedly entertain the people, how they COVID lab, COVID hospital, and diagnosis, and then infection, and then plasma therapy. We have no words. I am very grateful to forum and behalf of all the delegates. I hope the delegates are very happy to hear all our presence. Then coming to my own people, that is Dr. Shashi Kumar from the day one, then my Dr. Shilpa Shetty, MC, I have no words, then Sharmila, Vishak, and my whole team, and Dr. Fatima, madam, I should not forget, and then the A1 solution, who made this program very successful without any hurdles. I'm grateful to all of them, and our own Deepak, who arranged all the setup here, and all my other colleagues. More than that, all the delegates who participated, actually more than 1,250 people are registered, because there is a YouTube connections, so everybody must be watching through the YouTube. So and in addition, here also I have to thank our Praveen Jacob, and the then one more is Arun Tejas, who helped me to send the message of more than 3,000 delegates. So with the help of each and everyone, we could success this virtual conference. And I'm very grateful to each and everyone who helped us directly, indirectly for the success of conference. Thank you very much.
Thank you, madam.
Thank you, ma'am. Thank you, ma'am, for those wonderful words of gratitude. I once again thank all of you. Thank you, one and all.
Thank you, sir. Thank you, Srinivas sir, Madhyasth sir, all of you.
Thank you.
Thank you. Thank you.
Thank you.
Harish.
Thank you. Thank you, madam. Thank
All
you.
the panelists and Dr. Srinivas.
Thank you.